Yeah. Okay, is that good? Can you guys see it now? Is it decent sized?
Yes.
Yeah.
Okay. I can so I'll be advancing the slides for Scott, Melissa?
No, he's going to take care of that. He
Oh,
didn't
good.
send
Okay.
us.
All right, fine. Then don't have a problem.
I'm trying to get back into my Zoom mode and it won't.
It's
How
probably the
about
little black bar at the top of your screen,
It says view.
or it says mute, stop video participants.
when I went to return to meeting? Okay, here I am. The things I'm learning today, and I'm dependent on, it's 9:06. Hopefully Scott will be joining us. If not, I will be off Zoom,
Okay.
and giving him a phone call. Okay, I think though we should get started, even though I'm still not seeing him. Good morning, everyone. We do have a special speaker today. He is called Scott Anetto, who will share the knowledge of botany and taxonomy with you. In today's guest co-trainer, we have Serena Fox. Serena, could you briefly tell us about the projects you're volunteering on and what you've learned from CE classes, etc., and when you graduated and everything else?
Sure. I was in last year's class, and since I graduated, I have sampled a couple little things, which someone told me to do, which I think is a really good idea. But mostly I've been working with the Native Plant Propagation Guild. I've done a couple of marsh restoration projects with them. They're really fun. And then the big thing I've been doing is co-coordinating the Farm Day event, which is unfortunately a little different this year. And we'll talk to you about that later on down the road. But it's normally a sort of a large event involving volunteers that teaches kids, a bunch of elementary school kids, about soils and plant and plants. For CE, I've been doing that. The CE is great. They're very specific, tons and tons of classes on like how to prune winter roses, old roses in winter in Marin County. And when you get that specific, you get really super useful, good information. I just went to and did another one yesterday about deciduous tree pruning. A lot of them are on Zoom. So I've been doing a lot of the CE, the continuing education, and it's really a great resource.
Hey, good morning, Scott.
Good morning. How are you this morning?
I'm so glad to see you.
Yeah, thank you. Thanks for having me.
And we're just going to do, Serena, you want to advance the next slide?
Sure.
And it's your turn to talk today.
Okay, today our guest speaker will be providing our class with an introduction to botany and taxonomy. And later this morning, we'll explore 3 more MMG volunteer projects. And then finally, we'll close the garden gate. So it's short and sweet.
Okay, and next slide, Serena, please. Today, our guest speaker will be
That
As
would
we
be
would expect, plants are a big component of EFG, which is earth-friendly gardening, if you recall. Scott will give you a brief introduction to botany and taxonomy. An entire semester would only scratch the surface of this huge topic, so you can understand what we mean by brief. Next slide, please. Before introducing our guest speaker, I want to bring your attention to several brief housekeeping items. Always remember to mute yourself at all times during the presentation. If you happen to hear or notice a fellow trainee who has not muted themselves, please assist the rest of the class by muting them, which you can find in the chat box. Scott will be presenting a PowerPoint, and we recommend turning your camera off until his presentation ends. I'm going to ask you, Scott, do you want them to go into chat for questions, or do you want to take questions as they come?
I would love questions throughout the entire presentation today. So however we want to frame that, if people feel most comfortable putting them in the chat, that's fine. And if maybe somebody can help monitor that for me. And then, yeah, if people want to either raise their hand, or that might be the easiest way. But yeah, if somebody wants to monitor raise handing, just to cut me off so I can stop and take a breath and answer questions and have dialogue, that would be fabulous.
that sounds good.
We will be taking a 15-minute break, Scott, but when you feel it's a breaking point, go for it. I will let our speaker decide whether, and he did decide that, so we're done. Scott is a farm advisor with UCANR, supporting a number of countless counties in the eastern part of our state. Scott has multiple degrees from UC Davis, including a master in weed science. Scott, come to my house. I live right near Mount Verdell. You'd have a blast. As a researcher with the university, Scott has published extensive research on controlling weeds and other non-desirable plants. We are very, very fortunate to have someone with Scott's extensive knowledge and experience with us today. Now, Scott, you have some newbies here with Zoom. So I want to share the screen with you. Do I have to make you a co-host?
Yes, please. If you could make me a co-host, that would be great. Then I'd be allowed to share my.
Okay, I'm going to stop sharing. Is that all right?
Yep. And let's make him a co-host. Where would we do that?
Let's see. So in the upper right-hand corner, or if we see my thumbnail across the top of the panel, how you have your people displayed, you should be able to click on the three little dots on my profile.
More. Okay. I have three little dots, live on Facebook. Blah, 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 break for. No, you're not on my. I don't have a picture of you. Let me see if I can get you up on a picture. Oh, there you are. Okay, three little dots, and mute, stop video, make co-host. Oh, you're so good, Scott. Thanks for walking me through this.
Probably happy to help.
So you should be a co-host now. Let's see what happens.
There we go.
Aha! I did it, son of a gun.
That.
Thank you, Scott.
Sure. A couple little
No problem.
oh, do the video channel as well. See everybody. Their beautiful faces or beautiful names. All right. Do you want me to go ahead and get started?
Absolutely.
All right. Well, gosh, good morning, everyone. Welcome, and yes, it's so wonderful to be here. I think this might be maybe my second year helping with the Marin Master Gardener.
Second year, Scott.
Yeah, so it's great to always get involved with other counties. I would love to come in person. So if you guys ever do in-person trainings, I would love to come in person and meet all of you. But this is definitely the second best opportunity. So yeah, so as was mentioned, my name is Scott Oneto. Been with Cooperative Extension for about 25. I think this is my 25th year. I actually got introduced to Cooperative Extension shortly after graduating from college at Davis, and learned about the Master Gardener training before I really knew what I was going to do in life. My wife and I found the Master Gardener actually at the California State Fair of all places, and we loved plants. I was a botany major. She was a biological sciences major, and we loved growing plants and were learning at that time. And we got to talking to the master gardeners, and we just fell in love with their with everything about them. Their personalities, their willingness to want to share information, and so we decided to take the master gardener training ourselves, and that was back in 1998. So yeah, ever since then, I just fell in love with this program. I love what they do. I think they're just some of the best people in the world. And so yeah, I welcome all of you. I want to thank you from being a farm advisor at Cooperative Extension. We're all doing the same thing. I typically help focus on the commercial side of agriculture, but you all make my life and all of my colleagues' lives in terms of advisors, our lives are so much easier because of all of you answering all of those great questions that come in from our own gardeners. So thank you with that. So today, as mentioned, this is I guess a very brief summary of introduction to horticulture or botany. I'm also going to talk a little bit about taxonomy and how we name plants, and hopefully give you some tips and tricks on how to identify plants as we go through. Normally, I do those in two separate classes, but I think we've been able to structure this in a way where you're going to get the best of both worlds in today's lesson. So, as I mentioned, please interrupt as we go through. This is a very large group of topics, and to squeeze them into a couple of hours can be challenging, and especially depending on what information you're bringing or knowledge you're bringing, yourself into this. Some of this may be brand new for some of you, and others may have different types of botany or horticulture classes in the past. So we all have these different backgrounds that we bring to the Master Gardener training, which makes it even more powerful. So please don't hesitate to interrupt, ask questions. Again, put them in the chat or raise your hand. And if you don't know how to do the raise your hand thing, I think you should see it on the little toolbar in Zoom at the bottom. So I'll try to keep an eye out for the raised hands, and then you can questions and by let's see. So I guess without further ado, let me go ahead and get started here. So you should all be able to see my screen. If you can't, please interrupt. So let's go ahead and start. So I'm going to try to, if you had the chance to read the chapter on introduction to horticulture in the Master Gardener Handbook, boy, if you made it through that, congratulations, pat yourself on the back, because it is just I mean, talk about a snooze. It's a really tough chapter to get through. I think, and it's no criticism to the author, but it's really, really thick of information. It's really hard sometimes to visualize some of the things that are trying to be described and covered. So I'm going to try really hard to focus on some of the more difficult aspects of the chapter. And really, my focus is throughout the presentation today, I want to try to kind of correlate everything to plant growth and development. So that's kind of my goal. Because as gardeners, that's what we want to do, right? We're typically trying to grow plants. So trying to better understand how they do that and the different processes that go into plant growth and development and how we may even manipulate plant growth and all the kind of things that I want to share. So maybe just a quick, I guess, definition of what are plants, and these are three. I would say probably we could all probably come up with other things that we might use to describe or define plants. But when I think of plants, I think of these three things kind of come to mind. They are definitely living organisms, but what makes them kind of different with us is they don't have the ability to move. At least not in the sense of getting up and walking around. And so we have as gardeners and as people that give advice to other home gardeners, this kind of plays a critical role in terms of finding the right plant for the right place. Um, so we think about climate, we think about microclimate, we think about timing of year of when we might grow plants. So they thrive because again they can't put on a sweater if it's too cold, or they can't move into the shade if it's more of a shade level. So kind of thinking about plant choices and plant placement is kind of falls into the definition of what is a plant and some of the characteristics you have to think about. Another interesting, I
Scott,
guess, yes,
sorry, can you move a tiny bit closer to your mic? It's a little muffled.
you bet. Let me make sure. Let me just double check that. Let me choose. I've got several
Oh
microphones.
my God, so much better. Perfect. You fixed it.
Thank you for saying that. I've got several microphones hooked up to my computer, so sometimes I have to remember to choose the right one. So so hopefully that's better.
So much better. Thank
Good.
you.
Perfect. Thank you for interrupting. So the second, I guess I would say, uh, unique characteristic about plants is that they have the ability to manufacture their own food. And so we'll talk about photosynthesis and how that works in a little bit. And then lastly, I think another kind of unique characteristic about plants that's kind of fun, especially as gardeners, is this ability to regenerate lost organs. And so we'll talk about how plants can how we can regenerate new plants from plant parts, or if we want to do asexual propagation, what that looks like. So all of that kind of the plant responses to injury we'll talk about in more detail. So, plants fall into this category of what we call autotrophs. If you kind of break down the term autotrophs, it basically just means that it's a self-feeder, so it's able to feed itself, meaning it has this ability to manufacture its own food, and it does that through, of course, this process called photosynthesis. And you've probably all are familiar with photosynthesis at some level. And so I kind of just use this as a kind of a really simplistic way to show photosynthesis. It's a very complicated chemical reaction that occurs in the plants, but in summary, it basically is plants have this ability to capture energy from the sun. So basically, they're taking light energy, they're capturing that in their cells in the leaf, typically, and through the bringing up of water, again, typically from the soil, and mixing it or combining with carbon dioxide that it gets from the atmosphere, it's then able to undergo this chemical reaction that results in a carbohydrate or a sugar. And in addition to that, we have, of course, a byproduct of that whole chemical reaction, which is oxygen. So the plant takes those three sort of ingredients, carbon dioxide, sunlight, and water, and undergoes this chemical reaction. And then it gets the sugars, that it uses for growth and development, and it has oxygen as a byproduct. So it's just a fascinating chemical reaction, and of course, we rely on that because we are not autotrophs, we're heterotrophs, and we require other things so we need to feed ourselves the carbohydrates for growth and development, and of course we respire and need to take in oxygen for our process. So it's a good symbiotic relationship, I guess you could say. Um, so if we look just a little bit, I guess, in terms of how plants are built, of course, we have all are familiar probably with some level of biology in terms of we know living things are built of these building blocks called cells. And so I kind of like to think of cells as basically like Legos. So, we have different colored Legos, we have different shapes and sizes of Legos. And when we put Legos together, we're building things, right? So we're building structures. Um, and so this is basically the next couple of slides will kind of illustrate that. And so this little video that's just playing is just kind of showing, what this is a picture of a root, but you can see these little building blocks. And these little building blocks, of course, are called cells. And so there's all types of different cells in plants, various, again, shapes and sizes. They have different, I guess you could say, jobs in terms of what they are doing, in terms of functions for the plant. Um, but they do have some similarities in the sense that, they have certain structures within them, like a nucleus where their DNA is, and they have a cell wall that basically is that outer portion of the cell that keeps all the contents in. Um, and then there's various what we call organelles, or just parts of these different things, different structures inside the cells that are undergoing, again, different processes for growth and development. And so, I mentioned it undergoes photosynthesis. Plants undergo photosynthesis, and that chemical reaction occurs in these organelles, or these little structures that are called chloroplasts. And so we typically see a lot of chloroplasts in the cells that are in leaves, for example. So that's just kind of an example of what cells look like. But as I mentioned, I kind of think of them again as building blocks or Legos, and they, kind of form together to build structures. And so when we group similar like cells together, so we get what are called tissues. And so as you can see in this little diagram here, it kind of illustrates that, where this is just a diagram of a leaf going from top to bottom with all of the stuff in between that leaf. So, the top of the leaf here is illustrated with these little building blocks or cells that all look the same, and they're all of the same type, and they're all formed together to form a particular tissue type. So in this case, this is the epidermis, if you've heard that term before. So the sort of outer covering of the leaf is referred to as the epidermis, and that's just a particular tissue type. And then as you move down the leaf, in this case, you see this next layer. It's kind of like a cake, right? A wedding cake where you have all these different layers, and each layer may, look a little different, taste a little different. And so this next layer is called the ground tissue, and this ground tissue is again typically similar like cells that have lots of chloroplasts that are undergoing photosynthesis, and so that's kind of their job. And then we work our way down, and we have these little round balls. These are a different cell type. These are kind of, you can see they kind of look like almost like they're pretty far spaced apart. And that's where, of course, carbon dioxide is being stored in the plant, again, for that chemical reaction. And so, so you can kind of just get the sense that there's this sort of organizational structure of cells in any plant part. So this is a leaf, for example, but we see the same thing in a fruit or in a root or in a stem. Anywhere that we're looking at the inside of a plant, there's this sort of organizational structure. And then the last tissue type that I want to point out here is this one right here in the sort of center of this diagram, which is called the vascular tissue. And it kind of looks, you could kind of, I guess, say it kind of looks like maybe a tree trunk in this diagram. But this vascular tissue is just another tissue type that is again made up of specialized cells that are again carrying out some sort of function for the plant. And it's really the vascular tissue is the only one that I'm really going to go into any sort of detail on because it's kind of the more fascinating one. And then, you know, organizationally, so I said, similar cells form together to make tissues, and then of course many tissues. In this case, all of these tissues form together to actually make a larger structure, or what we call an organ for the plant. So, different organs might be a leaf, or it might be a flower, or it might be a fruit, or it might be a stem or root. So those are the kind of the largest structure. And then inside that structure, we have all of these groups of tissues. So let's look a little bit at the vascular tissue and a little bit closer because it's kind of a fun one to talk about. As I mentioned, it's made up of sort of specialized cells or these certain cell types, and those cell types are doing some sort of function for the plant. And so in the case of vascular tissue, it's made up of two different types of cells. We call them xylem, which is, of course, the water conducting tissue. So water moves through the plant. So through a very sophisticated tissue type called xylem. And the other one is, of course, the one that transports the food or the carbohydrates in the plant, and that is called the phloem. And I put this cross section of, you might all probably recognize this as a celery stalk, but it's nice to look at this one because it's one of the few plants that you can actually look at a portion of a leaf. In this case, it's the stalk of the leaf, and you can actually see these round little circles, which are the actual vascular tissue. These are the xylem and phloem. So that's where water's moving up into that leaf and where carbohydrates are moving throughout the plant through the phloem. So if we talk about these just in a tiny bit more detail, the xylem and the phloem, as I mentioned, the xylem moves water throughout the plant. It's also moving minerals, so nutrients or minerals that it's getting from the roots and from the soil. It's also transporting those, but it's mostly moving water throughout the plant. Usually, it is one direction, so it's moving upward from the roots up into the stems and out to the branches, and then ultimately out to the leaves, and then out of the plant through transpiration. You'll notice that there's these two different little drawings here, and those just represent the different cell types in the xylem. There's basically two that we see, and they're basically, I would just kind of say that I kind of consider them as just two different sizes of pipes. We've all probably have worked with PVC pipe or conduit, doing irrigation projects or whatever it is. So you can kind of just think of these things as being basically that, where we have these first ones here, which are called vessel member, but they're basically just little short fat pipes. So you could think of them as like really large PVC pipes, so like 3-inch diameter PVC pipe moving lots and lots of volume of water. And then we have these longer, narrower ones, which are just small diameter pipes. So you could think of this as like 1/2 inch or 3/4 inch pipe. So these basically work just like conduit does. It's basically they just stack one on top of each other, forming these very, very long, continuous runs of conduit throughout the plant. And if anybody is, of course, ever siphoned gas or siphoned water from a hot tub or whatever out of a hose, basically these kind of work the same way. They stack one on top of each other, and once they're in place, you have these long continuous runs where you can move lots and lots of water over great distances very quickly and also very efficiently. And then the last thing I'll point out about xylem, and I'll show some pictures of this and actually show why it's so important in a little bit. But when they're dead, so xylem cells, of course, any cells don't live forever, right? So the plant's constantly making new cells, and it's making new xylem. When the xylem actually dies within the plant, it actually becomes the structural integrity of that plant, meaning it forms sort of the hardness or the rigidity of that plant. And I'll show how that happens in just a few minutes. And then the other tissue that's in the vascular tissue, the other type of cells is the one that we call phloem. And I mentioned this one conducts the carbohydrates or the sugars throughout the plant. And so unlike the xylem, where it just moves in one direction from, basically an upward direction from the soil up into the above ground portion of the plant, the phloem, as you see in this picture, it's moving carbohydrates in both directions. And so, if you think of just a plant, of course, oh, that makes sense. We have the leaves of the plant are where photosynthesis or where carbohydrates are being made. And then a plant needs carbohydrates for all sorts of things. It needs it to, grow a new branch. It needs it to form a fruit. It needs it to put energy or reserves back into its root system for, maybe overwintering or for just growth and development for reserves for next year. So it's moving sugars all throughout the plant, depending on where it might be growing or where it needs energy. And it does that basically on this, like it's a process called source and sink interactions. And basically it's just this pool for energy that happens within these phloem cells. And so it's happening, continuously all throughout the plant, but again, through this kind of conduit system where we have these pipes and we have this sort of sap with carbohydrates moving in both directions throughout the plant. And so I said that this is called the vascular tissue. So the xylem and phloem occur in this, what we call vascular tissue. And the arrangement, and I kind of showed it in that first picture when I showed the leaf where it looked like a little tree trunk inside that model. The organizational structure of vascular tissue is very specific depending on the type of plant. And the main, we'll talk about these two really large classifications of plants, and you've probably heard these two terms before, and you probably know a little bit about what makes them unique. But we have what are called these monocotyledonous plants, which are sometimes just referred to as monocots. But those are basically things like grasses. So, turf grass, anything that else a monocot might be something like a lily or a daffodil or an orchid. All of those things are what we call monocots. And the term comes from monocotyledon, which basically the cotyledon, if you remember, is that little seed leaf inside the actual seed itself. So inside a seed, we have these little what are called seed leaves, and a plant will either have one of those seed leaves, which is a monocot, or it'll have two, which would make it a dicotyledonous plant. And so that's this very large grouping of plants. They're either going to be monocots or they're going to be dicots. And so the dicots would be, of course, anything that's not a grass. So we're talking about a very large grouping of plants. And the other way some people refer to the dicots are the broadleaf plants. So these are plants that have leaves that don't look like grasses. So they're broadleaf, meaning it could be something like a tree or a shrub, any sort of, herbaceous plant that you might have in your landscape that doesn't look like a grass would be a dicotyledonous plant. And so in that seed, it would have two of these little seed leaves. And so the organizational structure of these for the vascular tissue look very different. And you may be asking, well, why does that really make a difference, right? Why do I care about where the vascular tissue is in these plants? Well, it gets a little bit interesting in terms of, how the vascular tissue ultimately is responsible for how plants grow. And so that's one of the main reasons for pointing it out. So I want to focus mostly on the dicots because they're the most interesting. The monocots, I'll show you a picture of what theirs look like, but it's really not that interesting. So we'll talk mostly about dicots. And so I'm going to use this carrot as an example. It's a member of the dicot group. So it's a broadleaf plant. And again, the quickest way to differentiate, again, if you're still trying to mentally try to, figure those two out, I would say my first question I always ask myself or tell people to ask themselves is, does it look like a grass? Does that plant look like a grass? If, in terms of its leaf shape and what that leaf looks like. So we all know what a leaf on a grass looks like. It's kind of this, very long, strappy leaf. It has the veins on the leaf are all parallel, and so we all can visualize that. So when you look at a plant and you say, does it look like a grass? So you should you should very quickly look at a carrot and say, that does not look like a grass. And so that makes it a broadleaf dicot plant. So now that we've kind of deciphered or decided that that's a broadleaf plant, this is what the vascular tissue or the cellular structure looks like within that plant. And so this below picture on your bottom left, this is a cross section of a carrot, meaning if I just come in here and see if I can put a pointer. There we go. So if we get a razor blade or a knife and we just slice right in half the carrot here, and we kind of look right down it like we're looking down like the barrel of a gun or the pipe. This is what it looks like in cross section. And so this is the outer perimeter of the root. And then as we go towards the center of the root, we have all of these little round cells, right? So these are all a particular cell type, and they all look very, very similar. So they're all basically doing probably something very similar for the plant. And in this case, in a carrot, these are all a cell type that are basically storage cells. So they're basically storing carbohydrates for the plant. So they're all very, very similar, doing the same thing. And then we get to the middle or the center of the carrot, and then we have a very, very different cellular organization, and they look different. And so this is the vascular tissue. So this is where the xylem and phloem occurs inside a dicot. So it's basically forms this little bundle or this circle right in the center. And if we want to really dissect this, you'll see you can hopefully make out there's kind of an X or a cross here. So there's a line there and a line there of cells. So these are the cells that are the xylem that make that X. And then these little tiny ones that kind of make up this little grouping here, this little grouping here, basically in the apex of the X, those are all phloem cells. And again, these are lots and lots of conduits of pipes that are all stacked on top of each other that we're going basically from, the bottom of the root all the way up the center here up into the above ground portion of the plant. So that's where our vascular tissue is in the root. And then when we get up to basically where the root meets the shoots, you can see this top part, the green part. This is what it looks like in the dicot. And so again, this is the upper left picture here on the screen. And so rather than having that vascular bundle or that vascular tissue in the center, like it was in the root, what happens is that basically it branches out. So you can kind of think of this vascular tissue as being like your forearm in the root. And then once it gets to your wrist where it meets the soil interface, where it then transitions to shoots or above ground tissue, it basically branches out or kind of, I guess you could say, branches. That's probably the best way to describe it. And so rather than having a single trunk or bunch of pipes going right up the center, then they basically kind of finger off. And each of those little groups of fingers then form these little bundles. And so you can see now we have a whole bunch of these random or these organized bundles that occur around the whole entire perimeter of the dicot stem. And that is now the vascular tissue in the stem. And so again, that's the same for every single dicot plant. It doesn't matter if we're talking, a small little annual broadleaf plant or a very large tree that's a broadleaf. All of the vascular tissue is organized this way. And within one of these bundles is, again, xylem and phloem. The xylem is the stuff here in towards the center or the inside of one of these bundles, and the phloem is all of these smaller little pipes on the outside. And I'll show a close-up picture of that in a second. So here's that vascular tissue in the dicot root. Again, I kind of pointed this out already, but we have this very clear X that makes up the xylem. And then in the apex, we have a bunch of little pipes for the phloem. And then we go to the stem tissue or the above ground portion of the dicot. And again, I showed that we have these bundles that go all the way around the perimeter of the stem now. And within one of these bundles, we have xylem and phloem. The xylem are all of these larger ones kind of towards the inside, and the phloem is all of the ones towards the outside. And now how about the monocots? So I told you they weren't nearly as interesting, and we're going to go back to the dicots here in just a moment because the story doesn't end there. It gets much more interesting. So with monocots or the grasses, this is kind of their organizational structure. So again, we have in the lower left here, this is the root of a monocot. And kind of similarly, we have in towards the center of the root, we have this ring. And these larger circles or cells are the xylem. And in between them, we have phloem. And so you can kind of see what that looks like in the root. And when we get up into the above ground portion, kind of similarly, they also branch off. And so, but rather it being kind of a very clear pattern where we had in the dicots, we had these bundles that were very symmetrical around the whole perimeter of the stem. With the monocots or the grasses, we get more of this kind of spattering of clusters. And so all of these little, I guess you could almost think of them as looking like rice grains here. So these little kind of clusters of cells here, these are little bundles of vascular tissue that have xylem and phloem in them, kind of scattered across the whole entire leaf structure. So that's what monocots look like. So again, doesn't matter what kind of monocot, they all look like this. For dicots, they all looked like the previous one. So before I talk a bit more about the dicots specifically, I want to talk just a little bit about the xylem in plants and how water actually is taken up by the plants. And because if you think about it, we have, as I mentioned, the xylem is one direction and it's up. So we're basically moving water from the soil up into the root and up into the stems, the trunks, the tree, whatever the plant is, out to the leaves. And it's this continuous column of water or many columns of water that go up the plant and do that. But it's all against gravity, right? So it's basically going up and there's no little pump in there that's pumping the water up. It's kind of a really remarkable process that we're going to kind of show you how that happens. And so if we think about that, where that vascular tissue is again, so the xylem, where the xylem is located, in the case of a dicot, it's in the very center of the root. In order for the plant to use that water, it's got to get water molecules that are out in the soil into the root, all the way to the center of that root, where it can then be transported up the xylem into the plant. And so this is kind of just a diagram to show how water molecules enter into the plant. And it can do it basically one of two ways. First, it can actually take water molecules into the cells. And so we've all are probably familiar with root hairs. If you think of a root, in this case, a carrot here, you probably won't be able to see it on this camera, but all along the perimeter of this carrot, I've got these little tiny little whiskers that are poking out. And those little whiskers are what we call root hairs. And root hairs are basically really large cells. They're individual cells that increase the surface area of plants. And so they're absorbing water and minerals for the plant. And so if we look at this particular root hair here, you can see that water molecules are actually being taken up by the plant cell through a process called osmosis. And so it's moving against basically a concentration gradient. So it's taking up water molecules. And then as you can see at the movement of these water molecules on this diagram, it basically goes to the next cell. So water is going to be pumped out of that cell, pumped into the next cell, pumped out of that cell, pumped into the next cell, and moved all the way across this route until it gets into the vascular tissue where the xylem is. So that's one way water molecules move. The other one is that it can actually move in this other section down here on the chart, shows it moving in between the cells. And so you can see the water molecules moving in between the cellular spaces. So kind of just weaving its way through in between those cell walls all the way to, again, the xylem. And I guess what I would like to just point out is that either one of these two processes is super, super slow. So, you may be looking at, taking minutes for, a water molecule to move across either one of these two different pathways to get to the xylem. But once it gets in the xylem, remember I said it was a very highly advanced, process for moving large quantities of water very quickly. And, again, just thinking back to this idea of siphoning, if you, siphon out of a tube, just think of how much water, continuously flows out of that. And the same sort of thing goes for the xylem. And so once those water molecules get into the xylem, rather than it taking minutes for it to move a very, very short distance from the soil to the middle of the root, now it can move from the roots all the way to, tops of, the tallest trees that might be hundreds of feet tall. It can move that in like a fraction of the time. And so, rather than it being possibly seconds or minutes to do this short little travel, now it can move to the top of the tree within a second or fractions of a second. So very, very quick movement. And so this little video I'm going to show you is just a 20 seconds long or so, but it does a really great job kind of visualizing this process of water movement. So I'm going to go ahead and start it. And I think the audio should be optimized to play. So let's give it a shot and see. Oops. I think I need to turn off my laser pointer. There we go.
Plants transport water and nutrients from their roots to their leaves through a vascular tissue called xylem. These are dead, empty cells which connect end to end, forming long, continuous tubes. Because water molecules carry a slight electric charge, they're attracted to each other. This natural attraction, known as cohesion, links the molecules in long, continuous chains. At the leaves, water escapes the xylem and evaporates through microscopic pores on the surface called stomata. As each molecule breaks loose, it pulls up another molecule to take its place. This continuous pulling tension extends all the way down the xylem tube to the roots. Here, the rising chain of molecules keeps pressure low, allowing water and nutrients to enter through osmosis.
So hopefully you could hear that that video okay. And I'm going to just pause. Let's see, I'm going to do this slide and then I'm going to pause. I see some things maybe in the chat, so I just want to take a quick little breather and make sure there's no questions on this portion. So you kind of got this idea of how water molecules are being drawn into the xylem, and then of course, again, that attraction of water molecules to themselves basically forms these long continuous chains. And so you can kind of get this idea that as water molecules are being used for photosynthesis, so it's basically using those water molecules as part of that chemical reaction. It's using and pooling basically that chain to pull more water molecules up that chain, these long continuous pipes. And then that water, of course, is also evaporating out of the leaves. And so that's also pulling it. And so there's a whole equation for this process. And, it gets a little bit complicated in terms of the actual equation itself, but it's called the water potential. And so it's basically a gradient, a pressure gradient, moving from the water potential gradient in the soil through the whatever the water potential gradient is in the plant out to the basically atmospheric water potential. And you can kind of see that described here. Um, and you can see some of the numbers basically going from what we often see in the roots, that are usually down around zero point 0.2 megapascals, out to the atmosphere, which is minus 100. So it's moving against this negative pressure gradient. And we in agriculture often use that. And I don't have it in my office. I would grab it if I did. But we have tools that we can actually, or equipment that we can actually use to measure the water potential in plants. And so, so some people in agriculture, often that are growing tree crops or even vine crops, vineyards, can take a leaf and they'll put it into this basically this pressure chamber, and the machine or the equipment will measure the force that is needed to basically move water in that leaf. And when we get that reading, we can then determine or show what that force is. And often it's a good indicator of whether or not a plant is needs an irrigation cycle or is potentially water or drought stress. So, so it's a really helpful tool in a lot of agriculture. Um, let's see. The other thing, I guess that I would also just kind of point out is we often see kind of this phenomenon, with plants from time to time, whether it's a house plant that we may have, forgot about and neglected, and it got a little dry and it gets a little wilted. Or maybe it's, a plant outside that again might be in a pot or in the ground and maybe is showing signs of drought stress. And this is basically a response to, again, that water potential where the plants don't have enough water in the xylem, or within the plant itself to remain rigid, because the water basically is filling up all of those cells in the plant and kind of making them turgid or rigid, and kind of keeps them kind of almost like a balloon and inflated. As water becomes scarce, water moves out of those cells, it's used throughout the plant, and things start to collapse. And so that's what we basically see when plants are wilting is, this either collapse in the actual vascular tissue themselves, where, right, we saw those long chains of individual water molecules, and they are held together um, by this cohesion, and they want to attract one another, but that force is only so strong. And so as water molecules become scarce in the plant, that tension becomes greater and greater, and eventually those water molecules pull apart and snap. And once we have those the snapping occurring, it breaks that continuous column of water, and then we have a loss of that pipe. That pipe doesn't work anymore, and so that's when you get to basically, you reach the point of past a plant just showing some water stress to where it actually reaches the point where it can't move water again in that xylem because they've all collapsed. So let's take a second to just make sure we don't have any questions. Let me see if I can pull up the chat. Looks like, is everything mostly, it was all about
It's all
audio.
about the audio. I think
Perfect.
there's one question that's a little off topic that you can maybe get back to at the end, which is what is your
Okay.
definition of a weed? But
Yeah, maybe we can get that to the end. Yes. Well, I'll say
okay.
this right now. My definition of a weed is a fairly broad one, and my definition is a plant out of place. And so that leaves a lot to interpretation, right? So, and I think it's by intent, because a plant out of place you get to decide what that plant is, and you get to decide what that place is. So, a particular plant might be a weed over there, and it may not be a weed over there. And I'll use a fine example. I have a gravel, basically a gravel side yard, at my house, and I like to have it just gravel because that's where I keep my garbage cans and all of that. For some reason, California poppies love to grow in this little gravel area, and so I hand weed all of those California poppies out of that area. So in that case, they are a weed for me in that particular spot. I love them and nourish them and grow them in other parts of my yard, and they are not considered a weed, but in that particular spot, they're a weed. So hopefully that answers that. Okay, so getting back to, I'm going to kind of move to this topic of where plant growth occurs. And what I mean by plant growth is specifically where in a plant does it produce new plant parts? It's getting, taller because it's making new stem tissues or new branches. It's making new leaves. It's making new roots. So the roots are getting longer. All of that kind of stuff in terms of making new plant parts. That's what I mean by plant growth. So where does that occur? If we take this tree, for example, where is, it putting on new leaves and new stems, new branches? Is it at certain regions? Is it sort of just everywhere? Or is it more random? And so we'll talk about that. Hopefully, it'll make sense in a couple of slides where that actually occurs. And then the other thing that I'd like us to think about over the next few slides is that, and we've probably either have done this or have seen this, and it may not have been a nail, maybe it was a sign or a barbed wire fence, but if we were to put something in a tree, say a nail, and say we put it at 4 feet off the ground. We came in here and drove a nail there because we wanted to hang something there, and we drove a nail in there, and then we came back, say 20 years later. Where is that nail? Is it at the same spot? Is it higher because, of course, the tree's growing? Is it lower? So kind of think about that instance as well as we go through these next few slides. So I mentioned what I mean by plant growth. It's basically producing new parts, and plants do that through this process called cell division. Basically, it's mitosis. So we have a plant cell. It undergoes mitosis, meaning that it just replicates. It makes a new cell. And when you make a new cell, many, 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 many times over, those cells elongate, they get bigger, and you have new stuff, right? So basically, the plant has to make room for this new stuff, and so it does that through the formation of making new tissues and new organs. And so that's what I mean by plant growth. And so plants are doing that at what we call meristems, and they have two meristems that we'll talk about, apical meristems and lateral meristems, and that is where mitosis or cell division is taking place. So the first one is apical meristems, and these are found in all plants, and this is where we get all of those plant parts. So if you think about what makes up a plant, we've got this branch of this tree here, or this shrub, and we've got leaves, we have branches, we have trunks, we go down into the roots, we have roots. All of that is the result of apical meristems, the plant making new stems, leaves, flowers, roots, all of that kind of stuff. And these apical meristems are found at very distinct locations. So if we think of our dicots as an example, so these are our broadleaf plants again, they occur at the tip of the plant. So we have our tip of our plant up here. So we have our apical meristem. We also have them at the tip of all of our roots, and we also have them at all of the lateral buds. So you think of a plant, and you kind of go down the stem, and you see those little the little knobby things, which we call lateral buds. Those are also all apical meristems, and they're responsible again for growth. We all kind of know that, right? With grasses, we also have apical meristems. They're just located in the above ground portion in a little bit different location. So, rather than, having a tip of the plant like we do in broadleafs, with grasses, the apical meristem or the terminal meristem is down here, kind of tucked into where all of those leaves are again formed and where they eventually elongate and come out of. So the little apical meristem is tucked down in there. And so, if we think about, mowing a grass or a lawn, we come in here and we cut the grass and, mow it all off. We're not cutting the apical meristem. We're basically just cutting, leaves, leaf tissue, right? We're just cutting leaves in half. And so, if we think about what happens next, those leaves that we just cut, they're not going to grow back, right? They're not when we come back in 2 to 3 weeks to mow the lawn again, it's not those grass blades that we cut that are growing. It's all new leaves that are formed at the apical meristem that are being formed down here, and then they're elongating and getting taller, and that's the new grass that we're then going to cut. And then we basically repeat that cycle over and over and over again. So here, I love this example. This is, of course, Brussels sprouts, and what's cool about Brussels sprouts is it has huge lateral buds, which again are apical meristems. And so all along the plant here, we have these buds, and that's all those buds are apical meristems. And so what's nice about it is, you can cut into one and you can actually see, inside of a bud, which is sometimes hard on other plants when the buds are super small. So this is what you would find if you were to cut open a Brussels sprout. And so I'll pop one open here in a second, but what you see is that you have in the center of the bud is this little cone, and this cone is where mitosis cell division is taking place. So the plant's just undergoing cell division right here in this little dome. It's just undergoing cell division, making new cells. And as that plant makes new cells, those cells differentiate. Basically, they turn into whatever that that cell is that we talked about. Like, oh, it's part of the epidermis. Oh, it's part of that ground tissue in the leaf that's undergoing photosynthesis. It's part of the vascular tissue. It's xylem. It's phloem. All of that cell division, whatever cell differentiation, whatever that cell is going to become, all happens very quickly. All right here after it undergoes mitosis. And if we look as we go down, these cells get older. So it's undergoing mitosis. Those cells elongate. They get bigger. And as they elongate, it pushes that cone up, right? And it keeps making new cells and it keeps pushing that cone up, keeps pushing that cone up, keep pushing that cone up. And as that cone gets pushed out, you start getting other stuff, right? So let me just show you, for example, this plant here. And so here is let's see if I can hold these down just to make it clear. And this might be small for the picture. I'm going to go like this, and I'm going to stop share for one moment. Hopefully that'll make my screen larger for you all. Does that make the screen larger?
Yep.
Perfect. So so we have a terminal bud right here. Hopefully you can see. So that's the apical meristem. And as new cells are being made, they elongate, and it just keeps on pushing that up. So it just keeps on pushing this branch further and further and further out. So at one point in time, this little apical meristem was way down here attached to the side of this plant, but it just keeps on getting pushed out, and that just makes that branch and all of the leaves that fall out behind it, that's what all is coming out. And so these leaves here are fairly small, but it's because they just fell out of this bud, and over time the cells inside them are going to continue elongate and get bigger, and eventually they'll be the full size of the mature leaves. They just haven't reached their full size yet. They're basically still growing and developing. They're not undergoing any sort of cell division here. They're just basically still elongating and expanding. And you can kind of see that with the go back here. We can see that with our apical meristem here. So again, all that cell division is happening as we go back in time. Here's the formation of one leaf that just formed. We go back a little bit further. Put my laser pointer back on. We have a little bit older leaf. We go back in time even more. Here's a little bit larger leaf. And that's when you start seeing them actually fall out of the bud. And so no cell division is happening way down here inside these leaves. They all were formed up here. So all the cells already in them, they just need to expand a little bit more. And so we see the same thing in roots. It's just, of course, in a different location. This is a tip of a root. And so just back from the very, very tip, we have this sort of protective cap on each root. But where the dark kind of meets the light, again, we have this kind of dome area. And similarly, it's again an apical meristem where it's just undergoing cell division, making new root cells. And so as those cells elongate, it pushes that root deeper and deeper into the soil. So that's what those apical meristems are responsible for, is just adding new tissue, adding new tissue. Yeah, I'll come back to that sample in a second. Okay, let's see here. So I'm gonna go to the next one, which of course is the other type of meristem, and this is the lateral meristems. So apical meristems, I mentioned, are responsible for adding new plant parts. So adding new leaves, adding new stems, every single thing you can kind of think of in terms of parts of a plant comes from these apical meristems. The other type of meristem is the lateral meristems, and these are only found in woody plants. So woody plants being trees, shrubs, vines, those kinds of things that increase in girth, so get thicker. And the lateral meristems are responsible for that thickness, that increase in girth in plants. And they only, again, occur in woody plants. And if you think about those two large classifications of plants that I talked about, monocots and dicots, the only type of plants that can actually become woody out of those two are the dicots. So not all dicots become woody, but if a plant is to become woody, like a tree or a shrub or a vine, it's a dicot. So lateral meristems only occur in certain dicots. And so let's talk about how and where that occurs. So you may recall a picture that kind of looked similar to this earlier, and this is what the vascular tissue looks like in a dicot stem. Remember that picture of the dicot stem? I showed you the carrot, and I showed you where the orientation of the vascular tissue, the xylem and phloem, were. And basically they form these little bundles around the perimeter of the stem, right? So we have the stem and they have these little bundles that form around the perimeter. So when a dicot is going to get woody, so in the case I'm going to use this shrub again, and we'll make this a little bit bigger here in a second. When a plant starts to become woody, we get it from the vascular tissue. And so we have our little bundles of vascular tissue, our xylem and phloem. The xylem is, remember, the inside of these little bundles. That's represented by the red portion of these little circles. And then the blue portion is the phloem, or the food conducting tissue. And so when a plant is starting to become woody, what happens is that in between the xylem and phloem, the cells in between there become meristematic, meaning they start undergoing mitosis, right? So cell division. And so when cell division starts occurring, it's going to start making basically a ring is formed. And so it creates this meristematic ring. And so once you have a meristematic ring and we start undergoing mitosis, think of the plant replicating and making new cells. It's making new cells to the inside of this line. It's making new cells to the outside of those lines. The plant has to be able to make room for those new cells. And so what happens is that ring begins to move out. And so this is, you can kind of see the separation that happens between those original bundles, we have the little red section, the little blue section. We have that line that was created, which is the lateral meristem. And these are all the new cells that are being made on the inside of that line and new cells to the outside of that line. And the type of cells that the lateral meristem is making is new xylem and new phloem. That's it. It's not making new plant parts like leaves and stems. It's making xylem and it's making phloem. And that's basically what it looks like. So this is just a cross section of a branch, and where we have this transition. It was stained to be able to show the differentiation, but this line between the light and the dark is that lateral meristem. So to the inside of that lateral meristem, all of this light colored tissue would be xylem, and the red tissue would be the phloem. And so that's what is responsible for that thickness. And so again, as that lateral meristem continues to add new cells to the inside, it has to make room for them. So it just keeps on moving out, keeps on moving out. And that's what gives us that increase in girth over time. So just a little bit close up of what's happening with that. Again, kind of pointed all of this out, but again, here's that vascular, the vascular tissue or the lateral meristem, and it's making new xylem to the inside and it's making new phloem to the outside. Now, if I go way back to what, almost an hour ago when we started, and I spoke about xylem in general, I said one of the things that happens with xylem is that when it dies, it becomes part of the structural sort of support or integrity of a plant. And so if you think of a tree, for example, that has become woody, so think of a really large diameter tree, think of all of that really hard, dense wood that's in the center, or what we call the heartwood. That is all dead old xylem, right? So it's all xylem tissue. And then the sap wood, that's the stuff towards the outside, right? That's the actively growing stuff on the outside. That is basically where the phloem is, or the food conducting tissue. That's where the sap's, moving, and the carbohydrates, and that's right there underneath the bark, really, really close to the outer perimeter of that tree. And that's where it's moving all of the carbohydrates, right? So that's why we have to be so careful when we're, how easy it is to potentially girdle, right, a plant, a tree, or a shrub when we damage underneath that bark where that sapwood or where that vascular tissue is. If we disturb it too much, that's basically the life support of that plant. That's where it's actively moving carbohydrates and water is in that sapwood or in that vascular tissue that's right there underneath it. And so, the active part of the vascular tissue, the xylem and phloem, that's actually moving carbohydrates and active water is really close to this line, right? So, all of this, although it's all xylem, this is all the old xylem. So these cells are dead, they're no longer moving any water. So that's why it's really hard. And, when you cut a tree, you're not seeing any sap right there towards all that, hardwood. All of that's dead stuff. The actual active cells are the ones really close to the meristem where they were just formed, and then they conduct water for a while and conduct food for a while, and then they die, and then the plant makes new tubes, and those are the active ones. And so it's just this continual process. And so if we just think about that for a second a bit more, this is just a close-up of that line, the vascular tissue in that lateral meristem. And so it's making new cells to the inside, which are, again, xylem. I mentioned that. It's making new cells to the outside, which are the phloem. If you look at a chunk of wood or, a stump, you can then start to tell a story, right? If we've all probably seen or have counted rings in a tree before. And if we actually think about what we're counting, is we're actually counting portions of the vascular tissue. And so let me just explain that really quickly. If we look at this section right here, so this is all xylem, this is all old xylem, and it was all made from, of course, the lateral meristem. So it's in response to what the plant needs in terms of, again, moving water. And so when we have a plant that's, say a plant that is just coming out of spring, spring's starting to happen. There's lots and lots of water in the soil available. It has a high demand for putting on new leaves and growth and new stems, and so it needs to move a whole lot of water. It's going to be putting it, that vascular lateral meristem is going to be putting out lots and lots of big tubes, right? Remember that the big wide conduit tubes, the 3 inchers, those are all right here, right? So that's springtime. And then as we get into summer, water start getting a little bit more, scarce. And so we start seeing the plant making a mixture of big tubes and little tubes, right? So we have a mixture of big tubes and little tubes. We get into later summer, even less water available, less demand for growth. There's just less growth happening. The plant's making more little tubes, right? So we see less big tubes, more little tubes. And then we get into fall and winter, plant's still growing, but it just doesn't need to move as much water, so it's making mostly little tubes. And then spring comes again, the whole cycle repeats itself. And so we have this sort of transition from big tubes to little tubes, big tubes to little tubes. And so that is sort of that repeating pattern that we see when we're looking at counting rings on a tree, is just that repetitive, continuous pattern of the xylem making big tubes to little tubes over time. And so you can, again, use that as diagnoses for, being a master gardener. If somebody was to bring in, a plant or a tree that maybe died, and you wanted to, ask for a section of it, or maybe you did a site visit or a house call, if you did those kinds of things, you can look at a tree trunk and you can start telling a story of, how old the tree is, but you can also see, what's the distance between those repeating patterns of big to little xylem cells. If you don't, if you have really, really big sections, there may be a really active growing year, lots and lots of water available. But when they start getting really constricted, or it's mostly all small ones, maybe it was a drought year, maybe the plant didn't get enough water. All of those kinds of things can show up as part of maybe diagnosing or telling that story.
Scott, is this a good time to take a little break?
Yeah. So maybe I think before, I guess, is there any questions? This is a great stopping point. So yeah, if there's any questions on that section, I'd be happy to entertain.
There is questions in the chat.
Okay.
And I just lost.
I can read them. First question is, when do apical cells stop reproducing? A Brussels sprout only gets so big. What is the mechanism for stopping division, or does division just go on and it's too small to notice?
Good question. So it kind of depends. And I hate to use that. That's such an easy cop-out, right? So there's lots of things that are happening. We're going to talk about some of them a little bit later on. There's what are called plant hormones that help regulate plant growth and development. So we'll talk about one in particular, which is auxin, which does a lot of regulation for plant growth in terms of how big it gets, what buds are free to grow and which ones are being held basically dormant. So that's one thing that we'll talk about. Some plants are determinate versus indeterminate. So some plants are just like predisposed, kind of like we are, to get to maybe a certain height, right? It's part of their just genetic makeup. So we think about some tomatoes, for example. We have certain tomato types or varieties where they are determinate. They'll only get basically a certain height. Doesn't matter how much water, how much nutrients you get them. They basically reach a certain height and they just stop, and that's just kind of part of their genetic makeup. There's others that are indeterminate and they'll just keep on growing. They're like a weedy vine, and they'll grow 20, 30, 40, 50 feet long if they have the resources to do that. So I know that didn't completely answer your question, but those are some of the things that help regulate that.
Okay. Does this discussion have anything to do with keeping soil, compost, and fertilizer away from root crown to allow for better growth? That's from Stephen Fisher. I'm not sure which part that was in reference to.
Yeah, good question, Stephen. It doesn't. This discussion doesn't specifically have anything to do with keeping those three things: soil, compost, or fertilizer away from the root crown. Yeah, I mean, you know, probably we could have a discussion on that in terms of, the microorganisms and other things that might potentially, lead to decay or uh, pathogens or anything like that. If that's I'm not sure if that's kind of the direction you were going about protecting the root crown, but maybe we could save that one for later if we have more time to dig in on that.
Okay. Robin has a question. The individual rings, are those dead previous meristems, or are they just the difference between past xylem from seasonal differences?
That second part, yeah. So it's differences between past xylem from seasonal differences. So the meristem ring stays constant and active, and it just keeps on getting pushed out. And it's just all of the differences in past xylem that you're seeing between those rings.
And then David just says, "This is fantastic, Scott. Thank you. This would make an amazing workshop too."
Thank you, David.
You got a fan.
That sounds like a great spot to take a break. So I guess you guys want to
How
take a
about
15 break?
we meet back at a quarter to 11? Does that work for you, Scott?
That sounds great. We'll see you in just a little bit.
Okie doke. Thank you.
Well, we should have stopped that recording. There we go. There's Scott.
Yeah, I stopped it. Don't worry.
Oh, you did? Okay.
And we're back to recording. Hi, Scott.
Oh, there we go. Good to be back. Hopefully everybody had a nice little break. Good opportunity to try to digest some of that material or unload it. So let's see. I think I've got my share screen back up. So we just talked about all of the meristems, apical meristems, where a plant is forming new plant parts, so new leaves, new stems, new branches, new roots. And then we also finished up with talking about those lateral meristems that basically are the thickening of the plant. And so I am going to stop share for a moment. I'm going to switch my camera. And so hopefully you can see my camera. And if it's not super large, I'm not sure how you have on your computer, you may have a gallery of everybody's pictures. If you want to just see my camera really, really large, if you go in your upper right-hand corner of your Zoom screen, you'll see a little button that says View, and then you can click on the speaker one, and it should show that. But I think when I did that, it showed the Marin MG training one. So let's see. So let me just see if that will work. All right, looks like that camera froze up on me. So I will revert to to this one. um, okay, so we just talked about those two meristems, and the reason I wanted to just hold this shrub up. So this is just a branch off of a shrub that I took, and I think what this shrub illustrates really nicely is that some of these apical meristems are starting to undergo active growth. And so, up here at the top, we have an apical meristem. So you can see all of the new growth that's happening. So tucked inside there is undergoing mitosis and cell division, making new parts, making new leaves. So some of those early leaves are starting to pop out here. And over time, they're going to fully expand until we actually have full-size leaves. So all of that is occurring right here. And then again, as it's undergoing mitosis, it's pushing that little apical meristem up, or that dome up, and then all of the new stuff starts showing up underneath, making these branches longer. So of course that one's just starting to form. So at one point that little bud was way down here attached to the stem. And similarly, this one over here was also at one point way down here attached to the stem. And so over time, it just keeps on growing, and it just makes that branch longer and longer. The other thing I was going to point out is, again, thinking about the lateral meristem, or that thickening of the plant, all of this plant tissue is super, super soft. So if you think about, what the vascular tissue looks like, it's probably those individual bundles all around the perimeter of the stem. So there's no woodiness here. But then as you get down to some of this older plant stuff, now the stem starts getting really, really stiff and starting to thicken. Then we start getting the formation of that lateral meristem. It's making that secondary xylem, which is that heartwood, the center part of the tree that gets that stiffness to it. And so you can hopefully visualize that transitioning that's going as the plant's making new xylem cells up here, forming new plant tissues, making new stems. We have that continuous piping that's going down, and then eventually transitioning to form that lateral meristem into that older plant wood. Sometimes it's hard to actually visualize that, all that's happening inside the plant, but it is just a really remarkable process. Okay, so let's move back over to our presentation. So going back to this, just kind of going back to that picture and thinking about where plant growth and development is occurring. Of course, again, it is occurring at those meristems, at those apical meristems. So whether or not it's the terminal meristem on all of these individual branches, and also if there's any lateral branches that are forming, that's where elongation is happening in terms of the above ground portion of this tree. And then, of course, we have the thickening happening wherever that formation of that lateral meristem has occurred. And then just going back to that whole idea of where that nail would be. So if we, again, put a nail at, I think we said 4 feet off the ground, it came back in 20 years, it would still be at 4 feet. And that is because once that tissue is laid down, the only thing that is moving laterally is that apical meristem, because it's getting pushed up over time. So at one point in time, this terminal meristem, let me put it back onto the pointer here. So the terminal meristem of this whole entire plant, this little bud way up here, at one point in time was way down here. And over time, it's just building, basically putting new cells below it, and it just keeps getting pushed up, pushed up, pushed up, pushed up. So whenever that was made, this wood or this tree that was made down here at 4 feet, it never moves horizontally or vertically up and down. It basically stays right there. The only thing that would happen over time is, of course, that thickening of that lateral meristem. And so, of course, that nail would eventually get engulfed by that tree and up over time. Okay, so moving on, I'd like to just cover a little bit about some of the major plant parts and some of their functions and some of their importance of the plant. And so we've talked a little bit about leaves already in terms of them being sort of these solar panels for the plant, collecting sunlight and then converting that through photosynthesis to carbohydrates. And if we look at the anatomy of a leaf, it's really composed of two main structures. The big floppy thing out there that is referred to as the blade, and then they often have this little stalk called the petiole that makes up the whole leaf. And then the other thing I would point out on this picture is wherever a leaf attaches to a plant, you have a apical meristem, you have a lateral bud, right? So that's in terms of placement of things. When a plant makes a leaf, it puts a bud on the stem there. And that's just a repetitive thing that happens. So wherever a leaf attaches to the stem, you have a lateral bud. Now, one thing that I'll point out is there's lots and lots of different types of leaves on plants. But one of the things to keep in mind is that what often sometimes looks like a leaf may not necessarily be a leaf. And so, or what might look like many leaves, maybe how I should phrase it, what might look like many leaves, like the case in this middle one, might just be a single leaf. And what I mean by that is, again, by definition, a leaf is where it attaches to the plant. There is a bud. And so in this example, I'm just showing two main groupings of types of leaves. Probably the easiest one is, of course, this one on the far left, which we just call a simple leaf. And, by definition, a simple leaf has a blade, it has a little stalk, and it attaches to the stem, and there's a little bud there. In the case of these other two, these are what we refer to as compound leaves. And again, it's the placement of a bud that distinguish what a leaf is. And so in this case, these two different compound leaves, they have a bud right here where the whole thing attaches to the stem. So that whole thing only has one bud, and it's right down there, right where that whole thing attaches to the stem. Likewise with this one that's called palmate leaf, kind of like this gets its name from like the palm of your hand. So it has these little leaflets or these little blades. There's no bud up here. It's just one single bud where that whole thing attaches to the stem. So this is one leaf, and this whole thing is one leaf. And of course, the one on the far left is a whole leaf. They're just different types of leaves. This one on the far left is what we call a simple leaf, and these are what we refer to as compound leaves. And this will come up towards the end when we start talking about taxonomy and how we actually try to identify plants. This is a characteristic that we might use. We may need to know whether or not is that plant a simple leaf or is it a type of compound leaf, and that's why I bring this up. The other thing that we often will look at with plants is how those leaves actually attach to the plant in terms of the arrangement. And so there's usually three main types of arrangements. This one is called alternate. It's probably the most common one. And you'll notice again here in this example, you have a leaf attached to the stem, and where it attaches to the stem, you can see, hopefully you can see the little apical meristem, the little lateral bud. And then we go up to the next leaf, and they basically alternate up the stem. So we have a leaf on this side, we go up the stem, the next leaf plops out on the other side, and they basically just sort of stair step up the plant or alternate. That's where it gets its name. And again, wherever a leaf attaches, you have a little bud there. The other type is called opposite. So again, rather than having a single leaf that alternates up, now we have two leaves that are opposite of one another. So in this case, we have two leaves attached at the single place on the stem here, and where a leaf attaches to the stem, we have a bud. So in this case, we have a leaf attached on each side with a bud on each side. So we have two leaves, two buds attached there at that what we call node at the stem. And so it repeats itself again. So we have two leaves here, we go up the stem, we'll have another two leaves up there. And again, that pattern repeats itself throughout the plant. And then we could have these plants that are what we call whorled, and that's where it has more than three leaves. at a particular node. And again, like the other example that I just gave you, where a leaf attaches to the stem, you have a bud. So if in this case, if we had 3 leaves attached at that node, we would also have 3 buds attached at that node. So those are the 3 kind of main types of arrangements that we see in plants. And so I don't know if you can see this example really, really well on the camera here, but this is an example of an alternate leaf arrangement. So I've got a leaf there, I go up the stem, and then I have another leaf there, and then go up the stem, and then this one's just super short. I might have had some injury that caused this leaf to grow here, but basically this one is alternating up the stem for this plant. So that'd be alternate leaf arrangement. So I'm going to move on to the process of plants being able to regenerate lost organs. And I think we've all maybe have tried this at some point in time, where we maybe have taken a leaf off of our favorite plant and we decided we wanted to propagate it. And so we, took off a cutting and we put it in some starting mix, and over time, what we hope to happen is we get this formation of roots or whatever the lost plant parts are. And so this whole process of being able to regenerate lost organs is called adventitious growth. And it's basically the ability of plants to regenerate lost organs. And so if we think about, what the plant's actually doing, it's basically undergoing mitosis again, right? So it's undergoing cell division, and cell division's occurring, and then it's making new cells that then differentiate into whatever that lost part is. Like in this case, it's forming new root cells. And it's hard to, I guess, we talked about where plant growth occurs. And we know that, forming new plant parts occurs at apical meristems. And so that's the only place it occurs. And so, if I take, what would logically make sense is that if I wanted to propagate this particular shrub, and one way, let me see, I'm going to put my camera back up here. There we go. Okay. Couldn't see my camera, so I couldn't see what I'm showing you. So if we have a leaf here and we know where it attaches to there is a bud, logically, if we came in here and, wanted to start this plant, we could say, oh, we'll just, carve off this little section of stem that included the little bud. We put that in soil. All of that makes sense. There's an apical meristem there. We know what apical meristems do. They, replicate. They make new cells. A new plant forms. Okay, that all logically makes sense. But what if I told you I could take this same plant, and rather than including a meristem, say I just went like that, and I put this leaf in some starting mix, and it formed new roots. Now it's like, whoa, how does it do that? There's no apical meristem there. Well, that's the process of this adventitious growth, meaning what happens in the plant is I mentioned this idea of plant hormones, and we'll talk about them in a little bit in more detail, but there's several plant hormones in the plant that are responsible for dedifferentiating cells. And so meaning that if a plant gets injured and it loses a part, in this case, it lost a lot of parts, but these hormones will gather along the injury site and they will dedifferentiate. So whatever those cells were doing, they basically go back into a meristematic state, meaning they want to replicate and just undergo cell division. So these cells dedifferentiate, they become meristematic, they start undergoing cell division. Cell division occurs, it makes a new root apical meristem, and then once the new root apical meristem kicks in, it just starts doing its process, and then you have this sort of mass of of growth there, and then it starts forming all of those parts again, and then next thing you know, you have a new apical meristem that's making new stems and new leaves, and it basically replaces all of those lost parts. And so that's kind of this idea of asexual propagation. It's basically creating new plants from asexual plant material. So, and this is super, super common. Of course, we do it in horticulture. So if you want to share, say you have a house plant, or maybe, you have a coleus plant that you want to just, take a little snip off and root it and then share it with a friend or a neighbor. We know that happens. And in agriculture, they take it to a whole nother level. You think about any variety of, say, an apple variety. Say they're, Fuji apples. They create new Fuji apples by asexual propagation, and they do that by basically thinking of, the process is they, take tissue. In this case, they take a bunch of leaves from the Fuji apple and they stick it in a grinder or a blender, make a bunch of juice, and pour that little juice droplets onto petri dishes. And basically, again, the plants will de-differentiate those cells, undergo mitosis, and the whole process of apical meristems form, starts undergoing cell division, and you start a whole new plant out of that sort of callus tissue. And so that's how you keep making sure that you have Fuji apple trees and Thompson seedless grapes and Meyer lemons. All of those things are all genetically the same, because they're all created by asexual propagation to make sure there's no difference amongst individuals. Okay, so that's kind of the thought behind this idea of adventitious growth. I'd like to next move on to just talking brief. Let's see, maybe I'll stop to see if there's any questions on that. Yeah.
These clones, make these apples clones?
So, all right, so was the question, are these basically clones?
Yes.
Yeah, so
Yes.
these are all clones, correct. They all share the same exact genetic material, so they're all, in that sense, clones of one another. Any other
Scott,
questions? Yeah.
there's a question in chat. Is there an ecological advantage to having compound leaves?
Oh, an ecological advantage to having compound leaves. I'd have to sit on that one. I can't think.
I'm not sure if Robin meant an evolutionary advantage or an advantage to the ecology, like in terms of climate.
Yeah.
Robin, maybe we can.
Yeah, I was referring to whether, just in general, if maybe increasing surface area was actually giving them more of an advantage for having more photosynthesis or something along those lines.
Yeah. Never thought of that. Yeah, certainly I think that could be an advantage of just having, increasing surface area. Yeah, because I think of compound leaves. I mean, of course, they vary in size and shape as well. So sometimes I'll see plants with very, very small compound leaves. And in that case, I wouldn't think there would be necessarily an advantage over just a simple leaf of the same exact size. But then, there's some plants with very, very large compound leaves that might be, you think of something like tree of heaven, if you're familiar with that plant. Each one of those huge things that we often think of as being maybe many leaves, it might be 1.5 feet or even sometimes close to 2 feet long, that makes up an individual leaf with all of those individual smaller, what we call leaflets, attached. So yeah, I think that's a really interesting question, Robin. I just never really thought of it that way.
Also for the like succulents, it's a different kind of leaf, which actually makes it more able to withstand dry weather.
Yeah, certainly adaptations in the type or shape of leaf for dry weather, if that's what, the audio cut out just a little bit, but I think that was
Yeah,
your point was
yeah, yeah,
different plants might adapt either leaf shape or size due to
yes,
climate. Yeah, that's a great
right,
point. Yeah, I mean, you think of, I think the most extreme that I might think of offhand would be something like, a cactus, right? So, the individual spines are modified leaves. And so they have gone like, the complete extreme in terms of changing that leaf structure to something that is super, super resistant to hot, dry climates. All right. So I guess moving on to flowers, and I put the term up here, inflorescence, only because um, you know this term inflorescence is kind of the botanical term that's given to the whole entire flower structure of a plant. So, loosely we think of, we use the term flowers, and, a flower might be like an individual solitary flower where it's just, you think of like maybe a poppy where it makes, a stalk and there's one flower out there. That's a type of inflorescence. It's called solitary. There's other inflorescences where it might be kind of a spike, or maybe it's a cluster of flowers that makes up that type of flower structure for the plant. So basically, inflorescence refers to the entire flower structure for that particular plant. And so, of course, with the diversity of different types of plants, we have lots and lots of types of different inflorescences.
Scott, folks are seeing just the prezo. Do you want them to be seeing side by side of you, or
Oh, let's see. What are they seeing? I'm sorry.
they're seeing just the presentation? Do you want them to switch to the side by
Oh,
side view, or is it
they can
okay?
do the side by side. Yeah. They can go back to the side by side. Thank you.
So folks, under your view, if you choose side by side speaker, you can see both the, because Scott's holding some things up to his camera sometimes and
Ah,
gesturing. And
yes.
it's useful to see both, I think.
Thank you. So, I guess getting to what one of the functions or main, I guess, purposes of flowers is really the ultimate goal is to undergo fertilization for the plant, because that's its process of ensuring that its genetic material makes it to the next generation or its offspring. And so plants, undergo various different types of enhancements or modifications to increase fertilization, depending on how they're fertilized. So, some plants might be just wind pollinated, some might be self pollinated. Others might require an insect like a honeybee, or maybe some type of a bird or a moth to undergo fertilization. And so this just shows an example of one particular plant. Of this one in the case is pollinated by a bee or an insect, and it has what are called nectar guides as a color reference for attracting pollinators. So, what the bottom yellow flower might look like to us, for an insect, it looks more like that, where the reproductive structures of the flower are much darker and basically serve as kind of a landing pad or an incentive to visit it. So if we just spend a little bit of time looking at flower parts, and I apologize, I do this class in person. I love to have lots and lots of plant materials that you can kind of play with and dissect as we go through this. But I'll try to hold some up here as we go through and try to do the best I can, kind of showing you just from some of the pictures here, some of these different parts. But when we think of the different parts of a flower, I'll just point out some of the most common ones. And the first is what the stalk of the flower is called, and that's just what we call the pedicel. And that's basically where everything kind of takes off from, right? So the stalk is basically just holding the flower up. And so right above the pedicel, we have this little tiny kind of swollen area, which is called the receptacle. The receptacle is basically where all of the plant parts attach to. So if we think about all the major parts of a flower, like the petals and the sepals that we're going to talk about next, they all kind of attach to this receptacle area. And we'll talk more about the receptacle in just a little bit, because some of the fruits that we often eat are um, really formed out of either portions of or entirety of the receptacle tissue. So if we go out from the receptacle, the next one would be the sepals. And so these are typically, I would say, green in color for a lot of plants. It's sort of the outermost part of the flower. It's that part that basically encloses the flower bud before it opens. I think, one that just always kind of comes to mind when I think of flower buds with sepals, like a rosebud before it opens up, it's all encased. And then as that flower opens, those sepals kind of reflects or fall back. So they're often, again, green in color, and they're pretty conspicuous for the most part in a lot of flowers. The next one in would be the petals, and of course, I think that's pretty common for most. Um, and then we start looking at the reproductive structures. And so that's where we see the male structures, which would be the stamens. And then we have the female part, which is the pistil. So those are pretty much the main components of an individual flower. Now, the one thing I guess I would preface with that is that not all flowers have all of those parts. And specifically when it comes to the reproductive parts, we have some plants that will have flowers that have both those male and female parts. So both the stamens, the male, and the pistil, the female, and those are referred to as perfect flowers. And the opposite of that would be where they are lacking or missing one of those parts. And so that's what we would refer to as an imperfect flower. And we can even take that a little bit step further. So if it has just the male parts of a flower, we'll sometimes hear the word staminate flower. So they may just be called staminate flowers. That means they're male flowers. Or we might refer to them as pistillate flowers or female flowers. And we can take that whole kind of concept and take it one step, I guess, further, and actually start talking about the sex of the plant. So there's two main terms that we often use to distinguish the sex of a plant. So the first term here is monoecious. And if we kind of dissect this word down, we've talked about mono before. But mono is one, and ecious is house. So these are the one house plants, and these are those plants that have both the male and female parts on an individual plant. And I use the word plant there because, as you'll see in a second, I didn't use the word flower because there are some plants that may have male and female flowers on the same plant, but not in the same flower. So it's still monoecious because it still has both sexes on the same plant, but it could be made up of imperfect flowers, male flowers, female flowers, all on the same plant. And folks may be able to think about what some examples of that might be. I think the one for me being a gardener, I think of, all of the things in the cucurbit family. So squash and, cucumbers and melons. All of those plants have male flowers and female flowers. So both of them being imperfect, but they do occur on the same plant. So it's still monoecious. Likewise, we can just have plants that have perfect flowers, both male and female parts in all of their flowers, and those, of course, are monoecious. And then the other one would, of course, be dioecious, and that is, of course, di mean two, and then again, ecious being house, so these are the two house plants. And so those are where we have distinct male plants and distinct female plants. And of course, there's several different examples of that. I think kiwis being an example. Again, there's some weeds like tree of heaven, asparagus, several other examples that that have that as well. So as I mentioned, sort of that end goal of flowers in general is to undergo fertilization, and that's basically, you know, pollen from the stamens, from those male parts, being those pollen grains or the male gametes being um, uh, deposited onto the pistil, and then the male gametes making their way down to where the female ovules are at the bottom of the pistil for fertilization to occur. And it's kind of an interesting process. The pollen, those little grains of pollen, they land on the pistil, and then they actually form this tube that goes all the way down the length of the pistil down to the bottom of where the ovary is and the ovules are. And then once that tube is in place, the male gamete is then transferred down that. And so it's just a really remarkable process for, you know, to think of this pollen grain that's so tiny. It actually grows this little tube all the way down the length of that that pistil. And then, of course, once that male gamete's transferred, we have fertilization that occurs, and then once fertilization occurs, that flower will then start developing a fruit, and inside that fruit, of course, the result of that fertilization is the development of a seed, and so then we have a seed inside that fruit. And so here's just kind of a little character, I guess a little fun video of that pollen grain taking off from the male stamen, and those pollen grains land on top of the pistil. And then, as I mentioned, each of them have to grow this little tube that goes all the way down the length of the pistil to the bottom of the ovary. And then those male gametes are transferred down to where the female gametes are, and then fertilization occurs. And so just a really, really cool, remarkable process. And then, as I mentioned, the end result of that, all of that, is the formation of a seed, which is basically has this plant in it that is the combination of genetic material from both the male gametes, wherever that came from, and the female gametes in the pistil, wherever that came from. So it's going to have the genetic material from both of those. And inside that seed, of course, is that little seed leaf that I mentioned early on, the cotyledon. So remember, plants will either have one of those cotyledons being a monocot, so it's a grass or some, you know, in the kind of grass group, or it'll have two cotyledons, and that will be a dicot, or a dicotyledonous plant, or a broadleaf plant when it gets up and going. And we probably all have, you know, started seeds at some point in our lives, and you know, when the seed germinates and those little leaves pop out, those little seed leaves, you know, they often look very, very different than the actual true leaves once the plant actually starts making its true leaves. And so those are those cotyledons that I'm referring to. So just kind of a couple of fun things when it comes to flowers. You know, I like this example because I think so many of us are fond of this group of plants, the sunflower group. Often it's the Asteraceae family. And yeah, I think, you know, when we look at these flowers, and I use the term flowers because this is a type of inflorescence, and it's what appears to be a flower is actually many flowers. And what I mean by that is that when we look at a sunflower here, and I'll grab an example here and try to go through it as I show you. So I've got, these are mums. So hopefully people are familiar with this, but it's just, again, another type of plant that's in the sunflower family. Now, if I take off this and just show you what I'm referring to. So of course we have what we call petals around here. And if I pluck off one of these, so I pluck that off, that is a flower. this whole thing is a flower. This thing that I just plucked off is an individual flower. So there are hundreds and hundreds of flowers here. And to take that even further, so these are all flowers, and these are called, what do we call, ray flowers? These are a group of, or type of flower called ray flowers. Likewise, the center, all of this stuff here in the center, each one of those is also an individual flower. And those are called disc flowers. So again, I'll just go back to the screen here and kind of point those out. So these all along here with the big brightly colored yellow petals, each one of these, I pluck that off and it's got a petal, and inside that petal, if I look really, really close in here, and you won't be able to see it on this screen, but I'll try to get rid of my background and I'll just kind of use my pointer here. Inside there is a reproductive part. And in this case, these are staminate or male flowers. So these all have a little stamen in there. So these are all male flowers that release pollen. And if we go to the center of our sunflower, all of this dark stuff, and I kind of have a close-up here in the middle, or the bottom picture here, that's all that stuff in the middle. Each one of these is also an individual flower, meaning this is a cluster of petals all fused together to form this kind of tube. And if I break open that tube really carefully, there is a pistil in each one of these. So every single one of these is a pistillate flower, or a female flower. So it has both male and female flowers all in this inflorescence. So just kind of a fun thing with that group of sunflowers. And so if we think back at, sunflowers, if you've ever grown sunflowers or have harvest seeds of sunflowers, we think about where the seeds actually form or occur. It's all in that center part, right? So it's all in here is where the seeds occur. And that's, again, because that's where the pistils are. The female portion occurs in the center part. So I mentioned that once fertilization occurs, we get the formation of this fruit. And just by definition, a fruit is the plant parts, or combination of plant parts that basically contain that mature swollen ovary, that bottom section of the pistil, and a seed. So inside that fruit, we basically have the seed. And there's lots and lots of different types of fruits. And I just kind of put this listing here because it's kind of fun to think about. There's a whole classification system on how fruits form and the different way in which they form, and the different plant parts, in terms of the type of plant parts that actually are used to form that fruit. And so, some of the ones that you have probably heard of before, of course, nuts is a type of fruit classification. Berries is a classification of fruits, but it's interesting, a lot of things that we call berries are not berries by how we actually classify them. So I use an example of that. True berries, in the sense of how they're classified, would include things like blueberries, cranberries, grapes, bananas, kiwis. There's lots and lots of different types of berries. And again, that's just how they form. And then there's another group called aggregates, which is another classification or types of fruits. And those would be things that are like the brambles, so blackberries, raspberries, strawberries. Those are all examples of aggregate fruits. And then you've probably heard of some of the other ones, like pome fruits, or things like apples and quinces. And then there's drupes, and then there's multiple fruits. And then there's actually several other ones. I just didn't include them because these are the more common ones and more fun to talk about. So just to give an example of that, I said that, these are kind of classified in terms of the different plant parts that make them up. And so I just put two of them up here as an example. So I have citrus on the left. So this is, of course, is a lemon, and lemons are in the berry group. So they're actually a type of berry. And I also put up here a raspberry, and it's not a berry at all. It's actually an aggregate. And I'll talk a little bit more about that here in just a moment. So I'm going to pull up an example of a strawberry here, just to kind of show you how these form, and that whole classification system of fruit. So this is a strawberry flower, and I like this picture because it shows in pretty good detail. We can start looking at some of those plant parts and visualizing fruit formation. So kind of in the back here, you can kind of make out the tips of all of the sepals. So we all recognize those little green parts. Those are the sepals. That's what enclosed that flower before it opened. And then, of course, we have the petals. The little white things here are the petals. And then we have this ring of structures here that have these like little dry erasers on them. These are all the stamens. So these are the male parts of the flower. So you can see they kind of form this ring of stamens around the perimeter there. And then we have all of these little smaller structures here in the very center. And these are all pistils. So these are all the female structures. So aggregate flowers or aggregate fruits all form from a flower that kind of looks like this, where it has multiple stamens, multiple pistils, all in an individual flower. And so if we think about a strawberry, we look at the strawberry, we can again see those same structures, right? So we have the sepals here, the green structures, you can see the petals here. And then sort of the next thing in I kind of showed was that ring of stamens, right? And so if we look really close, if you ever, next time you pick up a strawberry, look really close in here where the sepals kind of touch the fruit, and you'll see little tiny whiskers in there, which were the stamens. And then the next, of course, structures that we talked about are those pistils, that whole group of pistils in there. And the pistils are basically all of these little things that we often sometimes loosely call seeds, but these are all of those sepals that were out here. And if you look again really closely next time you have a strawberry, you'll notice that attached to these little brown structures, you'll see the little tips, the little tubes on top of the pistils. And so if we cut open a strawberry, I'll just do that because I have one here in front of me. Grab a cutting board here. So, I said by definition, a fruit is a mature, ripened ovary containing seeds. And so if we cut open a strawberry, we cut open what we think of as a fruit, we should be seeing seeds in there. It's a mature, ripened ovary containing seeds. And of course, we all know that if I cut into a strawberry, which I just did, there's no big seed in there, like an avocado seed or, like a tomato has seeds in there, whatever the case may be. There's no seeds inside there. It's just all this like tissue. Well, what is that tissue? That is the receptacle. So remember that receptacle, that kind of little bulbous part of the flower where all the plant parts all came and attached to. Basically, it was, right there, kind of going back to the slide, that kind of swollen area on the stem. Well, that's the receptacle. And in the case of aggregate fruits, they form by basically filling that receptacle full of sugary basically carbohydrates. And as that receptacle fills up, I'll go back to the picture here. If you can kind of visualize the receptacle on the back side of this, where all of this attaches to, is it's filled up with sugar, kind of like a balloon. It's just kind of filling up that balloon. and it's pushing all of these pistils that were attached to the surface of that balloon out to the surface. So all of the actual seeds or the fruits are all on the surface. And so technically, going back to our little strawberry here, by definition, all of these are actually the individual fruits, because they inside each of these little brown structures would be a seed. And so same thing with with blackberries and raspberries. They form the same way, right? So we cut into a raspberry or a blackberry. You don't see seeds inside the actual structure. They occur on the surface of the fruit, or what we perceive to be the fruit. The only difference is that with the blackberries and raspberries, those little structures are juicy. They're they have juicy little fruits on the structure rather than the dried structure as a strawberry does, but they form the same way. So hopefully you'll never look at a strawberry or blackberry the same. Any questions on that little section?
Nope.
Okay. So I guess I'll just move to this idea of fruit formation, and what I'll, I guess, as an example here is, so say you have a gala tree, a gala apple tree in your yard, and your neighbor has also a gala apple tree in their yard. And if you were to pick that fruit, you would expect to get a gala, right? You have a gala apple tree, and you of course get pollen from a gala other tree. You would expect nothing short of a gala. What if your neighbor has a Fuji apple tree, and Fuji pollen gets on your gala tree, and you pick that gala fruit? Are you still expecting to get a gala apple tree or gala apple fruit? And the answer should be hopefully yes, because the fruit and the formation of that fruit, the taste and the texture, all of the things that we think about that makes a gala fruit, all of that is completely dependent on that parent plant, on that tree. It doesn't matter if it was pollinated by another gala. It doesn't matter if it was pollinated by Fuji or a Granny Smith or a Red Delicious. All of the characteristics of the actual fruit itself is from the parent plant. With that said, though, if we were to plant the seeds from those two different apples, they would be completely different. It wouldn't be a gala apple tree, and it wouldn't have gala apple fruit. It may kind of look, or it kind of might taste similar, but it's not going to be the same. And that's because the seeds contain the genetic material from the parent plants, right? So both the plant that it came off of, that fruit came off of, plus the pollen that contributed to the fertilization of that female to make the seed and ultimately the formation of that fruit. So even though the fruits taste the same, look the same, all of that, it's the seeds that would actually be different. And so going back to my earlier example of why asexual propagation is so critically important, especially for maintaining clones of some of these different plants that we grow, specifically, named varieties. It's because we want to keep that characteristic. So we ensure that by creating clones that are all exactly the same. Okay, so I'd like to transition into plant hormones, and I guess maybe I'll take a quick. Okay, no questions in the chat. I'll keep. If anybody has any questions, please, put it in the chat or.
Can I ask a question?
Yeah, please.
So I read Botany of Desire years ago, and I understood from that if you're going to have if let's say you have an orchard, you want to have a gala orchard, you would just take grafts from a tree to keep your consistent gala apples. And that, in fact, if you let your orchard grow wild, or if you didn't do that, you would have it would mutate, so you wouldn't have a recognizable, if an apple were to fall from that tree and fall to the ground, and result in a tree, it would not. This is what explains it. This,
Yeah.
because it's probably cross pollination. But if you had a whole orchard of apple trees and a bunch of apples fell off, all gala apples, wouldn't you? I guess still you could
They
you
would
would
have
still,
pollen. Yeah, there's even genetic variation within themselves. So yes,
yeah.
they would, you would basically continue to lose those characteristics.
So
And maybe
that's
you get something. Maybe you'd get something better, and that's all about plant breeding, right? So that's what we've been doing for millennia. Been plant breeding, selecting for those characteristics or those traits that we
right.
want. And so, you potentially would lose those characteristics that you originally wanted, either for better or for worse.
So therefore, if you wanted to have a gala orchard, or you really just wanted to grow Fuji's, you would do it off of a
Cutting.
graft.
Yes, exactly. Yep.
Gotcha. Thank you. That helps.
Yeah, sure. Okay. So I guess we're going to finish sort of this botany section on this topic of plant hormones. And I mentioned these earlier on, and these are basically chemicals inside the plant that help regulate plant growth and development. And there's 5 sort of major ones that I'm going to talk about. It's auxins, gibberellins, cytokinins, ethylene, and abscisic acid. Those are the 5 major players. And so these are basically produced in the plant. So the plant is producing these compounds and then moving them throughout the plant to have basically to help, with these different things that we've been kind of talking about, helping with, helping with as a defense mechanism. So, keeping, if you, need to start, recovering from an injury, it may wall off that injury. So we make a pruning cut. Plant hormones rush to that pruning cut and wall it off to keep out pathogens. A fruit falls off the tree. Those things are responsible for helping seal that off. A plant has a particular shape or structure. These hormones kind of play into that. A fruit might get really big. The hormones have a role in that. So let's go ahead and talk about some of those, because some of them are just super, super interesting. The first one I wanted to talk about is this one here called auxin, and some of the sort of broad things that auxin does. First, it is stimulating elongation of those cells. So I mentioned when cells replicate in mitosis, they eventually elongate. Auxin plays a critical role in that elongation process. They're basically, making sure those cells expand to their full size. Some of the other things as a result of that is auxin will direct stems towards light. So we've all seen, plants maybe in our house that might be in a, somewhat shady spot, and there might be a window somewhere in the room and they're kind of all bending in that direction. Auxin is responsible for that. The other big thing it does is suppresses branching. It's called apical dominance. I actually have a slide on that. It prevents fruits from dropping, so it keeps the fruits from, basically hanging on to the plant. And then another slide that I've got is talking about at low concentrations, we actually use this material or this compound to induce adventitious formation, right? So if I want to propagate a plant, I have a leaf from my plant that I want to propagate, I might dip this into auxin and put it in my rooting media, and that'll help stimulate that. So I can actually buy rooting hormone, and rooting hormone is basically synthetically made auxin. And then at really high concentrations, it basically kills plants, a certain group of plants. Broadleaf plants are very, very sensitive to high levels of auxin. So again, we can buy synthetic products that are auxin herbicides, and they basically kill the plant by it growing itself to death. Just think of lots and lots of cell elongation, shoot elongation. The plants start curling and twisting and cupping, and they just have no idea what to do with all that auxin. They basically just kind of grow themselves to death very, very quickly. So that is how that is used, and we'll talk about that. Okay, so here's just an example of what auxin is doing. Of course, we've probably could imagine what's going to happen. You lay a plant on its side, it naturally wants to bend up. Why? Because auxin is made in the tip. Gravity causes the auxin to settle on the bottom of that stem. More auxin on the bottom means more shoot elongation or more cell elongation. The result of that, of course, the bottom grows faster than the top of that stem, and so you see the plant bend towards the light, or bends upward because of that more elongation along the bottom of that stem. So that's, again, because of auxin. I mentioned apical dominance. So what I mean by that is auxin, again, is made at the apical meristem, or in these buds, the actively growing buds. And so at the very tip of this plant, we have lots and lots of auxin. And high levels of auxin basically suppress lateral buds. So all of those buds along this branch, we know there's lateral buds, right? Where a leaf attaches to the stem, there's lateral buds. Auxin keeps those buds in check, basically keeps them dormant. And so we call this apical dominance. Of course, some plants really have strong apical dominance, like a lot of conifers, for example. You think of the perfect Christmas tree. It has that very pyramid type shape, and that is because of auxin. Of course, we can manipulate plants in terms of their growth by altering the auxin levels, because, again, auxin moves through this plant basically by gravity. So we've got lots and lots of auxin at the top. As you move down the plant, you have less and less auxin being produced. And so, of course, what happens? You start seeing more lateral growth because there's less suppression. And by the time you get down to the bottom, very, very little suppression. So those branches are really, really big. And so, of course, what happens, say somebody comes in here and prunes off the top of this tree, or maybe a lightning strike hits the top of this tree. Something happens, takes the top of this tree off. What happens is one of two things. One, either one of these branches will say, "Ooh, I want to be the dominant leader," and it all of a sudden just kicks it into high gear and starts growing really fast and bends upward, and it becomes the new central leader. And then you basically get this new auxin dominant stem that's then releasing oxygen and keeping everything else in check. And you basically kind of see almost this pyramid on top of another pyramid. Or you see basically just the release of growth from that cut stump. So you have a little cut here, and those buds right below it just say, "Woohoo, we're not being suppressed anymore," and then they start growing. And so we see that all the time, and we can manipulate that. So I'm going to just show, hopefully, this example as an example of that. So this is a tree in my yard. This is a Photinia bush that just, it gets massacred. I mean, I chop this thing all the time for classes and stuff. So, I want to point out right here in the center is a pruning cut. So I pruned this last year, probably for a Master Gardener class. And if you think about the leaf arrangement, remember we talked about leaf arrangement. In this particular plant, I mentioned this has alternate leaf arrangement. So there would have been a leaf attached right here, and where a leaf attaches, there's a bud. Go down to the next node, there would have been a leaf attached here, and where a leaf attaches, there's a bud. And so when I made that pruning cut last year, there was probably just a single leaf here and a bud, a single leaf there and a bud. And when I made that pruning cut, I removed the auxin source. And so that released this bud and this bud to start growing. And so, of course, what happens? It undergoes mitosis. Now that bud is way out here. Same thing. This bud that was there is now way, way out here at that tip. And so that released those buds. And so if we want to make a plant bushier, especially like, something like, gosh, I think of, a lot of my herb bushes, like herb plants that I grow, or, like a coleus plant, anything that you want, something that often gets like leggy because it's not putting on lots of lateral growth. We can totally manipulate it by going in and tipping, basically pinching off those terminal buds where the auxin's produced, and then that releases those buds below that to then grow to fill in that plant to make it bushier. So that's all that we're basically doing. And then I mentioned, of course, it can be at low concentrations. We buy it as a rooting hormone. So Rutone is one example. There's lots and lots of different ones. And these are just the chemical names of those, but they're all basically auxin compounds. And similarly, I mentioned at high concentrations, it's a broadleaf herbicide. And there's tons of broadleaf herbicides. Basically, every broadleaf herbicide that's a selective broadleaf herbicide is an auxin herbicide. So this includes, current chemicals like 2,4,5 or 2,4-D. So it's that's a common herbicide for like lawns to control broadleaf plants in your lawn. They think of something like if you use brush begone, so triclopyr products, 2,4,5-T, which no longer is available. That was Agent Orange, MCPA. There's just lots and lots of auxin herbicides that again have been formulated. The second one I want to talk about is gibberellins. This one's also kind of a fun one. This one is responsible for a couple of things. One, it stimulates shoot and fruit growth, and it also helps with seed germination. And so I think probably the interesting example with gibberellins is, again, it occurs naturally in plants, but it also is used commercially in agriculture to make fruits really big. So probably the best example of that would be with Thompson seedless grapes. It's often applied, so it's called gib acid. Gib acid is often applied early on just after fertilization occurs. So just as you're starting to get the formation of a grape, it is applied to the developing clusters, and basically it's kind of fooling the plant to say, send more sugars, send more sugars. And so that's what makes those fruits often really, really large, is the addition of gibberellic acid. The third one is the group of cytokinins, and this one does a couple of things. It also stimulates cell division, so it's important with the adventitious and asexual propagation. It also helps govern regeneration of plants as part of that. And then it also correlates root and shoot growth. So, often, if we think about a plant as a whole, the top often usually matches the bottom, meaning that the root mass is often similar in size to the above ground canopy. And so cytokinins is the chemical or the compound that helps regulate that. And then we have ethylene. So ethylene is an interesting one because whereas all the other ones are basically a liquid in the plant, ethylene is actually a gas that's moving around inside the plant. And this one is responsible for a couple of things. One, it's a ripening hormone, so it helps ripen fruit on the plant. And then also it's probably the most primary in terms of when a plant is injured. This is sort of the injury response hormone that helps kind of wall off and helps with some of those defense mechanisms. And I do have this YouTube video. I'm not going to play it, but if you're interested, you can take a screenshot of it or take a picture with your phone, or watch it another time. It's kind of an interesting video on how ethylene is used again in agriculture. This one particularly is, I think, all focused on the Dole company, mostly with banana production. And so how bananas are harvested, super green, super unripe, and then they go through a series of basically gas chambers or ethylene treatments to ripen them to eventually showing up on our supermarket shelves.
Scott,
Yeah.
I actually have a question about that. Is it true that bananas continue to outgas ethylene once you bring them home, and therefore, if you put other fruit next to them, it'll ripen it?
Yeah. A lot of fruits naturally off-gas ethylene. Bananas are a big one, and the other really big one is apples. And so, yeah, if you ever need to ripen other fruit, certainly sticking them in a paper bag with either overripe apples or bananas can really help stimulate or help with that ripening process.
And then Robin asks, is this one the cut grass smell? I'm not sure which one she's referring to.
Ah, cut grass smell. I mean, I think of like underripe bananas smelling like cut grass. Is that what you mean, Robin?
I meant because I've actually heard that grass, when you mow the grass, that smell that you smell is the grass like freaking out and telling other grass like, "Oh, we're getting cut." So I
Oh,
was just curious if that was like
interesting.
that ethylene is that
Oh, I
hormone.
wonder. I don't know. How interesting. I've never heard that, but I am going to make a note to look that up because that's super interesting. I mean, I totally. I mean, that's what happens. I mean, when a plant gets injured, ethylene is like the compound that gets sent to those to that signal. Basically, is the signaling device. I don't know if it has an odor. Um, I'm going to look that up. That's super interesting. Thank you for bringing that up. Cut grass and ethylene. So yeah, check out that video if you are interested. It's kind of interesting. And then the last one is this one here called abscisic acid. And I think this one's interesting because it was actually discovered at UC Davis, and it is responsible for a couple of things. It's another stress signal, kind of like ethylene, and it basically causes. And I didn't include the slide in this talk, but there was reference to stomates. Stomates are these specialized cells that occur on the surface of the leaf. And basically, they just open and close to help with lots of things. One, when they open, they allow carbon dioxide to come in. They also allow, of course, oxygen that was a byproduct of photosynthesis to escape. But then they're also allowing water vapor to leave the plant to be able to keep the plant cool, to kind of create a little microenvironment around the plant. So it helps with the regulation of that stomates opening and closing. And then it's also a dormancy signal in terms of, you think of plants that go dormant during the winter. There's actually what is called an abscission zone on plants. And so I'll show you, I think I've got a picture of that here. When a deciduous plant, so think of a plant that's going to, go deciduous in the fall and drop its leaves, there's a zone where that leaf attaches to the stem called the abscission zone. So these are, again, just this particular cell type. And when the abscisic acid concentrates in this area, basically it just kind of seals off or walls off this zone. And as it does that, basically it just walls off or causes that organ, in this case a leaf, to just drop or to fall off. And so whether it's a leaf or a branch, they all have these, abscission zones here. And so that's one of the reasons, if you think about, you'll probably talk more about, proper pruning cuts at some point. Way back in the day, there was a lot more people doing like flush cuts. So, if this was, a branch and you were going to prune this branch off, it wasn't uncommon back in the day to just try to get that thing as flush as possible. You bring in your saw or use your cutting shears, and you basically prune and try to get as flush cut as you possibly can. Well, now we know that there is this abscission zone, this naturally occurring area that is a defense area for the plant. Basically, it's going to wall off that. So if this is that same instance, that branch, we now cut at the collar. And that collar is basically that abscission zone where the plant has that natural cells that are designed to wall off and seal off in the case of an injury. And so we prune off, leave that little collar. The abscisic acid helps, and ethylene help kind of wall this off and prevent any pathogens, bacteria, whatever, from getting in there and infecting the plant. And so, of course, in the fall, when leaves fall, we actually see a leaf scar. And so that scar, of course, is just that abscission zone where that leaf was attached to the plant. And then the last thing I want to talk about before we transition to the last topic of today is this idea of tropism. So the hormones that I just kind of covered were all sort of these internal plant regulators. These are things that the plant is making internally to help with plant growth and development. This idea of tropisms is more of an external response. So not something that actually, the plant is necessarily doing, but maybe it's an external signal that's help regulating or modifying our plant growth and development. And so these tropisms might help the plant nutritionally, make them maybe move the roots to a particular location, like downward, maybe move the shoots towards sun to capture more energy from the sun. And so there's 3 different types of tropisms that I'll just quickly mention. And the first one is what we call phototropism. And you could probably tell by the name that it's basically the signal is the sun, so photo, and so it's a light signal. And so this is what causes certain plants to be more phototropic, meaning they bend towards the light more than others, is they just have this tropism that they want to try to bend or elongate their plant, the stem towards light. And so they use auxin, of course, and so the auxin basically concentrates away from the direction of the sun, and it causes those cells to elongate, and then that stem bends towards the sunlight. And so this could happen like on an individual day, right? So there's plants that track the sun across the sky, and again, they're using auxin as the source, but it's the light that's actually making the auxin move to, again, cause that cell elongation to occur. And then once the sun goes down, the auxin redistributes, the plants, the plant cells elongate the other way and go back, and it just keeps this kind of process going back and forth. It's kind of really fascinating. And then there's one that is specifically on gravity. So this is typically for roots, but this causes roots to grow downward. And so there's structures in the tip of the roots that are called, I think I just lost the name. Are they otoliths, or is that what's in our ear? I think they're called otoliths. I forgot. I apologize. I just completely forgot. But there's these little structures in the roots that basically kind of similar to auxin. When they concentrate on the lower side, they again cause cell elongation to occur on the other side. And so that's basically what's happening here. And then lastly is thigmotropism, which is a response to touch. And this, of course, there's a couple different like really common plants. One of them being this plant here on the left, which is sometimes referred to as touch plant. Very common in very tropic areas, but basically you touch it, or wind passes by it, an animal brushes by it, and it basically causes the leaves to collapse. It loses all that turgor pressure, the water pressure in the plant for a second, and basically causes the leaves all to collapse. And then eventually it'll perk back up, and you can just keep on doing it. And then, of course, the other one here on the right is a Venus flytrap, which uses it for nutrition. It's basically capturing its a food source. Okay, so we're wrapping up. We're getting kind of towards the end here. I've got probably another couple dozen slides that I'd like to get through, to hopefully get through all of them in the next half hour. So hopefully folks are still doing okay. So let's kind of transition over to plant taxonomy. And I'm going to kind of focus more on how I use taxonomy for identifying plants. Because really the idea behind plant taxonomy is sort of this hierarchical, this structure of organizational, of grouping plants and eventually giving plants name. And so I think why this is important for us as, master gardeners and giving advice is, I think a lot of people rely on master gardeners for just plant identification in general. It's like, oh, what is this plant I found in my yard? I just moved to the neighborhood and I have no idea what plants are here. Can you help me? And so I think that's a role that we often play. I think, you know, in terms of identifying any sort of pests or diseases on plants, we often have to know what that host is, what the actual plant is. So it all comes back to plant identification. I think making good plant recommendations also falls into why it's important to know plants. And then I think, you know, identifying any types of poisonous plants also, you know, is helpful for us and also our clientele. And then, of course, we do use lots and lots of plants for food and medicine. And so being able to recognize which plants those might be also, I think, is a good tool. So, you know, I think there's an endless, this slide could be much busier in terms of how we identify plants, whether you use, you know, field guides or things like maybe the A to Z Garden Plant Encyclopedia or Sunset Western Garden Book, use websites. Now there's all of these apps that can be on your smart devices that work really good sometimes. And so, yeah, lots and lots of different tools for identifying plants. So if we look at the history, and I'll just make this one kind of quick, the system that we currently use, which is called the scientific classification system, started with Carl Linnaeus. And basically he developed this sort of classification system based on structure. And it's basically putting liked things or grouping liked organisms together based on similarities. And so if we kind of look at the overall structure of that, you probably have maybe seen this at some point. This is kind of what that organizational structure looks like. And it's basically going from broad to specific, meaning, you know, we have these few, typically, I can't remember how many there are now. I think in school they're teaching people that I think there's maybe 7 or 8 kingdoms, but these are very large groupings of organisms. So we have the plant kingdom, we have the fungi kingdom, we have the insect kingdom, we have several different bacteria kingdoms. And so these are very large groupings of living things. And then, of course, within, say, the plant kingdom, then we have a number of divisions. So again, very large categories like angiosperms and gymnosperms and monocots and dicots. That's where those large kind of classifications or breaking of large groups occurs up towards the top of this. And then as we move down this classification system, we start getting to some of those ones that we might use on a more regular basis, like, oh, what plant family is that in? Oh, that's in the sunflower family, or maybe that's in the legume or pea family. So we often use family, and then, of course, within family, there are genera, which is the genus. So again, it's even a more refined or closer to the individual species. And then we have individual species, which is the combination of the genus and that specific epithet. And I'll show you some examples of that. Hopefully that'll be a little clearer. So just to kind of have a rough outline of what the plant kingdom looks like. This is kind of just a real simple idea of what it might look like. So again, I plant kingdom at the top, and then I mentioned there's just a few divisions. We have, I mentioned 2 large ones being the angiosperms and the gymnosperms. And then there's a couple of other ones here called the pterophytes, the lycophytes, and the bryophytes. And so these are basically some of the things like ferns and the club mosses, things that are plants, but not really a lot of, you know, the plants that we see or deal with on a daily basis. So the vast majority of things that we're often working with are typically in the angiosperm division and the gymnosperms. But then you can see underneath the angiosperms, we have the monocots and the dicotyledonous plants. So you can hopefully start seeing, you know, how this goes from very broad, you know, characteristics. It's a plant, and then it's a flowering plant, the angiosperms, and then we have monocots and dicots, and it just keeps on getting more find more characteristics that are similar as you kind of go down this family tree. So as I mentioned, we're typically looking at these last three in this sort of hierarchy, being the plant family, the genus, and the specific epithet. And then, of course, we see this on, the plant tags at the nurseries and, magazines in terms of articles that we might be reading or articles that you might be writing. So whenever we're using, plant names, we're often sometimes either using or seeing this. And so I just put a couple examples up here. This is, of course, you're probably familiar maybe with this plant. This is purple coneflower. It's in the sunflower family. So you'll see the family sometimes listed on the plant tag, and it ends in one of the rules of this nomenclature. It ends with the letters A, C, E, A, E. So all plant families will end in A, C, E, A, E. So the legume family, for example, is the Fabaceae family. And so anytime you see those letters A, C, E, A, E, that's referring to the plant family. And then you may see, you'll also see the genus and the specific epithet. And those two terms together is what makes the species. In this case, here is that listed here. So Echinacea is the genus, and the second term, the specific epithet, is Purpurea, which is referring to the color. And so, of course, plant families have, again, sort of similarities in common. So they might have similar like shaped flowers, and they may kind of create the same type of seed or the same type of fruit. Their leaf arrangements kind of might be similar. So all of those kind of, I guess you would say, those morphological characteristics that make up the plant will often be characteristics that might be similar across family lines. And again, as I mentioned, they all will end in that ACEAE. And of course, some of the cultural practices within, in terms of how you might care for a plant, might also follow those family lines. And then the actual scientific name, the two words that make up the species, as I mentioned, is that first word, which is the genus. In this case, it's Echinacea. And then that second word is we call it used to be a long time ago, they just called it the species, but now it's called the specific epithet. And so that's that second term, which is Purpurea. And then it's those two words together that actually make up the species. So this particular plant is Echinacea purpurea. And that's how you would see that. And you'll notice that on the tag that the scientific name is in italics. And if it's not in italics, it would also be either, it would be underlined. So it's either italics or underlined. And the other, I guess, thing that I would also notice is you'll notice that Echinacea is a noun, and so it's always capitalized. And then the specific epithet is an adjective that's describing that noun, and so it's always lowercase. And so those are just some common rules, and those are just always the case. It's, so if you see scientific names, or you're going to write a scientific name, maybe for, some sort of a Master Gardener article, we try to encourage you follow this rule set where, again, it's capital for the genus and lowercase for the specific epithet. And then plant names, of course, can be further classified into some of these things, right? So I'll go back to this example here. We had Echinacea purpurea, and now we have all of these other types of Echinacea purpureas. This one you can see is the ruby giant. And what does that mean? Well, it might be one of these here, either a variety, cultivar, hybrid, or maybe an heirloom. And so how do you know what that is? Well, talking about the differences between them, I guess the first is that a variety is something that is occurring naturally. So it's basically a unique characteristic to that species that warrants it being called something different, meaning that maybe it's a different flower color, maybe the leaf looks completely different. So some type of unique characteristic from the original species. And so this is just an example of a variety. This, if you're familiar with this plant, or if you're not familiar with this plant, this is a type of redbud. So this is Cyrsus canadensis. And it's typically like the picture here, it often has these very vibrant pink flowers in the early springtime. But there is a variety, so this is a naturally occurring type of redbud that has white flowers. And so this particular variety was named Cyrsus canadensis variety. So when it's a variety, that'll spell out VAR, and then they'll give it some other unique characteristic name. So in this case, it's alba. So alba is Latin for white. So this is the white eastern redbud. And so, so that's where you'll see varieties. If it's naturally occurring, it'll be the scientific name, and then it'll say VAR for variety, and then another describer. Likewise, we can have what are called cultivars. So varieties occur naturally. Cultivars are basically a result of basically breeding. So, human intervention caused a cultivar. So this is a cultivated variety, is one way you could think of it. So, so this was bred. Cultivars are bred to have some, again, desired characteristics. So, in this case, you'll see that rather than it saying variety, we'll have the single quotes here. So this, in this case, this is a cultivar called forest pansy, and it's was given that name after these very, very dark burgundy leaves that form on the plant. So, so this is, again, still the same species, Cyrsus canadensis. It's an eastern redbud, but this particular cultivar is forest pansy. So going back to that echinacea, because it's in those single quotes, we know that ruby giant is also a cultivated variety, so it's a cultivar. So hopefully that makes sense. And then I want to do this one here, which is a hybrid. And so the hybrid is one where you get a unique individual from mating two individuals with basically different genetic makeups, meaning we may breed two different tomato types that don't naturally cross, but we can, do it to create a basically what is called a hybrid. And so a hybrid might be something that they might breed a hybrid for like increased vigor. So basically make lots and lots of tomatoes. So if anybody's ever grown like early girl tomatoes, I think that's kind of one that always kind of comes to mind, because I like to grow early girl tomatoes, and I do it because they're super, super vigorous. They grow really fast, tons and tons of fruit production. And so they are an example of a hybrid. And you'll notice a hybrid when you buy them or see them, is you will often, it'll be shown on the label as this notation of typically F1. F1's the common one. So it means that it's the first generation seed of that cross. And so I think the important thing to keep in mind when it comes to hybrids is that because they were formed from two dissimilar plants, they're when you grow them from the first generation seed, that seed doesn't always stay true. So sometimes you can save seeds, right? So sometimes you can save seeds from certain tomatoes and they'll basically stay true. You get the same characteristic. And that's not always the truth, because we've talked about that with the whole Gala apples. And, if you save seeds from an apple, you're going to get probably something that looks very different. So in often cases, we don't save seeds, but there are certain plants that do have a tendency to stay true. But hybrids often do not. So you can, grow you have to buy basically early girl tomato seeds, every year to have early girls. You could save the seeds, you could grow them, but it's probably not going to have those characteristics. And then the last one I think is this one here, which is an heirloom. And an heirloom is kind of it's not a real clear definition, but it's basically an old cultivar. So again, we learned what a cultivar. A cultivar was a cultivated variety. So some human intervention, bred a particular tomato in this case for a desired characteristic, and over time was able to stabilize it so that you could save the seeds and that same characteristic would be true. So you could continue to save seeds, grow those seeds, save those seeds, grow those seeds, and it basically those traits are stable. And so usually an heirloom is defined as roughly 100 years old, but some say, between 50 and 100. But so that's when you see like on a label, in this case, Brandywine tomatoes. If you've ever grown or familiar with Brandywine, these are a type of heirloom tomatoes. And that just means that it's an old cultivar that basically has stable traits, so you can save the seeds and you would expect to have, again, Brandywine tomatoes out of the saved seeds. And I guess maybe I'll just cover this one briefly, the whole thing on GMOs. And so basically GMOs by definition is an organism that has had, again, human intervention, but what makes it different from a cultivar or even a hybrid is that it includes using DNA from a different, typically a different species. It can be a completely different species or a completely different organism, or that they alter the DNA in a way that it wouldn't occur naturally by just by plant breeding by itself. So that's typically what is meant by GMO. And so there's about a dozen different GMO plants that are grown. Probably the two largest, I would say, would be corn and soybeans. I guess you could probably throw cotton in there as well, but it's not a food crop, of course. So yeah, there's lots of, I think one of the things that I'm not so happy about the whole GMO thing is often a lot of seed companies use it as a marketing tool. There's no GMO seeds that I know of that are available to home gardeners. And so I think the whole, where it's GMO free on seed companies, there's no GMO tomato seeds, so why would you even put that on the label? But I can see why it's used as marketing. Okay, so I'm going to just finish up with a couple of last slides, going back to this whole idea of hierarchy and grouping plants. I mentioned under the plant kingdom, there's two large divisions, again, the angiosperms and gymnosperms. And so hopefully you're familiar with these groupings of plants. They're pretty distinct, right? Gymnosperms are conifers. They make cones. They don't have conspicuous flowers. They don't make these fruits that we think of because their reproductive structures are cones. And so that's one, again, very large classification of plants. And then the other one, which I think is probably the most predominant, are these angiosperms. And these are, by definition, the flowering plants. They have, again, it's a huge group. So we're talking about the monocots and the dicots that fall within this group. So we have, this group of flowering plants, and then the two categories right underneath them is whether or not they're a monocot or a dicot. So tons and tons of different plant families that fall into this category. So both grasses and broadleaf plants. And so this just shows those two different breakdowns. So I mentioned within the flowering plants, the angiosperms, there's the monocots and dicots. We've talked a lot about some of these characteristics of how you tell them apart. I said probably the easiest is just look at the plant and does it look like a grass? If it looks like a grass, it's a monocot. If it doesn't look like a grass, then it is a dicot. Of course, by definition, we've talked about what that means. Monocots have one seed leaf or one cotyledon, and dicots have two. But the other characteristic that you can also use is the floral characteristics, looking at the flowers. And monocots will have their flower parts in multiples of 3. So like we talked about petals and we talked about sepals, monocots have 3 petals or 6 petals, any type of multiples of 3s, whereas dicots would have theirs in multiples of 4s or 5s. And just to give you an example of that, here's 2 flowers that you could kind of say look kind of similar, right? And so if I was to ask you, is the plant on the left a monocot or a dicot? I'm not showing you any leaves intentionally, but if you were to just count parts, you could go through and label these, just count them 1, 2, 3, 4, 5, 6. So because it has multiples of 3s, we would know that that's a monocot. If we look at this plant on the right, again, you can kind of see leaves in the back, but if we just count the floral parts, we have 1 petal, 2 petals, 3 petals, 4 petals, 5 petals. So we know on the left we have a monocot, and on the right we have a dicot. So we can kind of start using some of those characteristics to start kind of grouping plants based on where they fit in that hierarchy of nomenclature. Similarly, if we were to look at these 2 plants, hopefully you could recognize the one on the left as a type of conifer or a gymnosperm. Again, it produces its fruiting structures in cones. And then on the right, we have, this is a type of angiosperm. It's a flowering plant, right? So remember that first category is it an angiosperm, a flower, it makes some type of a flower, or is it a gymnosperm, it makes some sort of a cone? And so that's kind of similar with, I mentioned the difference between a monocot and a dicot. Does it look like a grass? The same question could be said. If it doesn't look like a conifer, then it's an angiosperm. And then right underneath an angiosperm is it a monocot or is it a dicot? Again, ask yourself, does that tree look like a grass? No. So it is an angiosperm and it is a dicot. So hopefully that kind of makes sense. I apologize, I went through some of those slides a little bit fast, but I wanted to get down to these last few. I wanted to just give some resources out there for plant identification. I've been kind of leaning. I felt like there was a pretty strong taxonomist, and I could key out plants pretty well using Jepson Manual and lots of other plants books for identifying plants. But more recently, I think I'm using my phone more for some of these. And so some of the ones that I use a lot, probably the number one that I use now is Seek. So if you haven't, downloaded a plant identification app yet, I would encourage Seek. It's not 100%, but it works pretty darn good. And then there's some other ones listed here as well. The one last thing that I'm going to show you, I've got like 2 minutes and I'll show it to you. I do have a dichotomous key that's available. And if you're unfamiliar with what a dichotomous key is, it's basically kind of the old school way of identifying plants. And it's basically a kind of a book formatted in a way where you're presented with 2 different statements, and you basically choose the statement that best represents your sample. For example, I might give you, if I had this flower here, and the 2 statements said flower yellow or flower pink. Well, I would obviously choose the statement that said flower pink. And then at the end of that statement, it would lead me to 2 new statements. And it's kind of written in this kind of like just style where you basically just choose the statement based on the characteristics, and then eventually it leads you to whatever plant that is. And so that's how kind of dichotomous keys are written. So, if you're interested, I have this book here that's available for free. You can download it. I think the PDF we did some modifications on our website, so I hope that link is still good. But I know the online one is still works. So it's available either as an ebook for free or as a downloadable PDF. And basically, it's kind of a watered down version of a dichotomous key for identifying plants for Northern California. And so I'm going to just spend 1 minute to just show you how it works. So, for example, somebody brought you in maybe a picture, or maybe they brought you in this particular tree, and they wanted you to identify it. So one, you could use an app, or you could use this book. And just to show you how this book works, here's just a close-up of the leaves. So it's got kind of these kind of roundish leaves with pink flowers. And if I don't know if you'll be able to read the detail, but I'll just read it out to you. Basically, it asks you these first two statements. We start off with 1A and 1B. 1A asks if this is a gymnosperm, or 1B asks if it's an angiosperm. And so, of course, it goes into more detail as to what each of those is, but hopefully you could just say, okay, we know that's an angiosperm because it's got regular leaves and it doesn't look like a conifer. And so if we chose 1B, you'll notice at the end of 1B, it tells you to go to statements 2. So we just drop down here to 2A and 2B, and it asks 2A, does it have leaves or does it not have leaves? Well, we have leaves, so we would choose 2A, and then it tells us to go to statement 3. On 3, it says leaves are simple, or 3B, leaves compound. Well, we learned earlier that this is a simple leaf. It has a blade and a petiole, and where it attaches to the stem, there's a bud. And so we would choose leaves simple, and then it tells us to go to statement 30. So we go to statement 30, and 30 says mature leaves and branches opposite or world. So again, we're talking about leaf arrangement and how, remember, there's alternate, opposite, and world. And then 30B says mature leaves and branches alternate. So again, looking at how these leaves attach to the stem, hopefully you can see how they stair step. So they're alternate. And so we would choose 30B. And then 30B tells us to go to 31. 31 says how many veins are in the leaf. And so again, we choose that it has more than one prominent vein. And so you can kind of see how this goes. And it just keeps on going through this process. And then finally, you get to statement 40 that says, it asks about whether or not the leaf is lobed or not. And it basically, as you can see here, it keys it out to redbud, Cirrus occidentalis. So that's kind of how a dichotomous key works. So it's not too terribly difficult, depending on the key, but super, super effective. So I can't believe I got that done exactly right on time.
Perfectly timed. 12:30 on the dot.
But yeah, gosh, I want to just thank you all for great questions, and it's just always great to be with Master Gardeners to spend the day. I'd be happy to entertain questions, but I know you guys also have an agenda to stick to as well. So you're always welcome to contact me offline too. I'll stick my email address in the chat, and so if you want to reach me offline, you are more than welcome to. Or if you want to ask a quick question, I could do that too.
David just put the link to the key
oh, perfect. Thank you so much,
in the chat as well.
And Scott, thank you. I'm gonna. I don't recall if I sent you Patrick's email, but we usually post your presentation for our trainees to review for their final.
Oh yeah, I'd be more than happy to send that on.
Okay, that would be absolutely great, and I will send you his email tonight.
Great, I will.
And I want to thank you once again for your great presentation. You had to cover a lot and very in-depth, and I just can't thank you enough because you've really touched the high points.
No, thank you so much. Yeah, I appreciate that. And yeah, it's always great to spend the day with with Master Gardeners. So thank you.
Thank you so
Thank
much.
you so much. Okay, Serena, it's up to you. Can you get us back to we're going to take a 5-minute, 10-minute break. So we're all going to be back here at 12:40, and then we'll finish up. And
40. Yep.
Serena, you going to do the honors with the slides?
Yeah, are you sharing right now?
No.
Okay. Well, let me see if I can.
I could if you want. It's up to you.
I got it. I'm just getting to the presentation. Here we go.
Okay.
And
Beautiful. I'm going to put us on pause. Serena,
yes, ma'am.
we're up to the overview of 3 MMG. I tied my hair back too. And you get to start it.
Are we good? Is everybody seeing the slide?
I do.
Okay, so Cinda is not able to join us today, so Melissa and I will be introducing another exciting subset of MMG projects, which are the plant sales. These are critical fundraisers for the organization and an important way to give back to the organization and to the community. And it's kind of fun. This first one is the pollinator plant sale. So these are pollinator attracting plants. The pollinator plant sales team coordinates the propagation and sale of about 40 different types of pollinator plants. The pollinators, we all know, are vitally important to the overall health of our ecosystem, and they're fundamental to the concept of earth-friendly gardening. And in addition to bee-friendly plants, the pollinator sale also has plants that attract local butterflies, flies, hummingbird species. This year's sale is coming up on Saturday, March 9th, which you're welcome to go to the sale. But work for each season sale usually begins about in October. And volunteers are needed in the winter months to help with tray and pot filling, seeding, and plant transport. The project is headed up by Tom Hammond and Nancy Oblentz. Any questions? Nope.
Okie doke. The tomato market.
Big deal.
Very big deal. So a couple of things. I find it really strange that you were asked to volunteer but didn't get a form to order the plants beforehand. So there's a very easy way to remedy that. You can go to VMS, and at the very, very top of VMS, there's a place where you can go and order your plants. So the tomato market is the MMG's largest fundraiser and serves a vitally important role in providing revenue to operate our many volunteer projects. This year's market is being held on Saturday, April 20th. The tomato market is the first project many trainees engage with us, as it is the first opportunity for trainees to begin earning volunteer hours. Excuse me. You have been offered a chance to volunteer since there was a change that you now can volunteer before you graduate. It is up to the people who are in charge, who is Peter Norton and Roger Pankoska, to decide what tasks they will give you. And that is true for every project leader when you volunteer as a trainee. This is a great way. Oh, by the way, the volunteers are needed to transport tomato plants from the greenhouses to the site of the markets. And they're hosted at Peony in Novato, as well as Bon Air Center in Greenbrae. In addition to tomato transporters, volunteers are also needed to staff the sale. This is a great way to support the organization and meet other master gardeners. There is no long-term commitment unless you'd like to become more involved with the growing and the shepherding of plants next year. I think, Steve, you had a comment about that. I saw it pop up. Okay, there are a few volunteer activities in advance of the tomato market prior to the sale. I assume these are on VMS and are eligible for credit hours. Yes, they are. And also, too, as I said, it's always up to the project leader to decide where you are going to volunteer based on the fact that you're a trainee. Once you pass the final and graduate, you can volunteer pretty much on anything. The basic reason for that is they want you to be certified when talking to the public. Any further questions about the tomato market? Before we wrap up with the final project, I've already told you about the pre-purchasing, and if we can go down to slide 11, Serena, that would be great. You can see on the VMS details about the tomato pre-sale are available on the VMS homepage. I used this yesterday. It worked perfectly fine. I didn't want to go through all my emails that are saved to find it. And you can find out the varieties, you can find out their characteristics, and then you can place your order. They are requesting that you prepay. That's a change from before. Before they used to say checks or credit cards, but now you just prepay on the computer. Okay, next slide. The succulent plant sale. Oh, I'm just looking at my notes, and Patrick is suggesting if you are considering purchasing tomatoes, please do not wait until the day of the market. I highly recommend ordering during the pre-order period through February 19th, as people line up hours before the market open, and we sell out early every year. And I can attest to that. For before I became a master gardener, I was at Peony's with my book, my phone, boxes, because the lines are unbelievable. So next slide about the succulent sale, and take it away, Serena.
I totally agree about that. The tomato sale is amazing. They sell out, all the plants go in like 2 or 3 hours. It's incredible. Succulent plant sale is an annual succulent plant sale that continues. It continues to grow in popularity, and it's another great way to get involved. Volunteers for this project grow succulents from cuttings, and then they foster them at home over the winter months, and then transition them to the Falkirk greenhouse prior to the sale. The sale's held this year on Saturday, May 18th at Falkirk in San Rafael. I don't know if everybody knows where that is. And the project is led by Lillian Track.
Okay. Okay. Can you move on to the coming up slide? Oh, we're on. Yep, Jay. choose plants with care. Think about everything Scott presented today and how that will reflect into our earth-friendly gardening. I just want to say, I know today had a lot of information. Don't be overwhelmed by it. I guess I'm trying to say, all we want you to know is that there's genus, there's families, there's hybrid, but just to be familiar with the terms. There are still times where I have to go back and look up, what does hybrid mean? I'll give you a little clue about the stamen and the pistil. How I remember, and someone else gave me this, the stamen, it always stays men. So that's how I remember it's the male part. And I think a pistil packing mama for the female part. And that's the only way I can remember how to do that. But again, please don't be overwhelmed. We just want you to be familiar with the terms and know that they somewhat exist. Okay, coming up, hopefully that's the next slide. Yay! First, I'd like to thank the 17 trainees who have already taken the January survey. The feedback provided has been very informative, and I plan to provide a high-level overview of the key themes next week. If you have not yet completed the survey, I hope you take advantage of today's class, concluding a little bit early to take the survey while you're at your computer. Please be aware the survey will close at the end of the day on Friday, but I think it will still be open, so don't worry about that. Homework 5 is due February 14th. We love sending you homework as a Valentine's gift. GPPD is due in class February 15th. Please bring your binder to class. Next week is going to be just an incredible class. That's all I can say. First of all, it's going to be about roses. Second of all, Rod has so many great things planned. He's actually bringing in about 20 rose bushes, and you're going to be pruning them. So, and I will be sending out reminders. Please bring clean pruning shears. Please bring your gloves, and I will bring the newspaper to cover the tables. I'll also provide coffee and hot water. We're also going to get a presentation about garden pests, mostly ones that afflict roses, by our co-vice president, Joan Kozlowski. And Cinda will be back next week to bring us 3 new exciting projects. And I think we are up to closing the garden gate. And as usual, I and Serena will stay on for a few more minutes if you have any questions. If you want to touch the past, touch a rock. If you want to touch the present, touch a flower. If you want to touch the future, touch a light. Hope you all have a great week, and I hope you enjoy what's left of the sun today. Thank you.
Thank you.
Thank you.
Thanks.
We've got chat popping up.
Thank you. Great class.
Thank you, Chuck.
There's a question about the presentation on the CT, and yes, the answer is be there.
Yeah, I sent them and I thought I put it, I guess I just didn't send it, Serena, but I thought I did.
Yeah, it's still there. There's just something. I guess it went out several times and I had huge gaps. So, I'll have to
It's,
watch that again.
yeah, it's gonna, and I strongly recommend, especially the section on taxonomy, because that's really interesting, and it's really it will play an important part, Olaire, in what you do in the future with master gardeners, being able for plant ID, etc.
Mm-hmm.
And like I said, which I thought I sent, Patrick is out of town, so it's not going to go up immediately. And I will be sending Scott Patrick's email. So I would look for it on maybe Sunday or Monday.
Okay. Okay, great. Thank you so much. I appreciate that. And you said we're supposed to bring gloves and pruners next week?
Yes.
Yes. Okay. Thank you.
I have a question, Melissa.
Yes, Katie.
There's additionally a 2.5 hour video that we are to watch for this week. And then there's an optional one that is 3.5 hours, which I figure is probably the same as what we just heard because it was recorded during the COVID time. So is that about right that I need to watch
that? Katie, your question, and I think I mentioned this last week, is forget the videos, pay most attention to your reading.
Okay.
The videos were geared during the COVID period, and also too, let's say you want to get information again after you graduate, you forget something, these videos will be here to help you get that information.
Okay. So that's true, even though one's marked optional and one is not, they're both optional.
I would say so. I mean, I just think, just looking at Scott's video today, he really did a really, really, really great presentation.
It was great.
And it has all the information I think you will need.
Okay, thanks so much.
You're welcome. And I think we're down to 13. Serena, thank you so much for helping me. I couldn't have done it without you.
No problem.
And good luck with what's the word I'm looking for? And usually by Thursday after this, I am totally blank. Good luck with your adventure with Farm Day.
Thank you.
I think you and Curtis are an excellent team for that project. I think you bring so much great warmth and enthusiasm to it.
Oh, that's nice. Thank you.
So hopefully I will see you soon, and you just take care, and thank you again.
You're so welcome.
Marty, are you still there? I don't see him. I think he left. So sounds good. I am going to head out and try to get my brain back.
All right.
Take care,
everybody.
Bye-bye.
Can you remind me how to sign up for the tomato market?